Good afternoon, everyone. On behalf of Krupanidhi Group of Institutions, I welcome you all to KrupaPharmaCon 2021. Krupanidhi Group of Institutions was founded in 1985 under the leadership of Professor Dr. Suresh Nagpal. Started with the center goal of nurturing as many young minds in the country as possible, we've gone global in this drive with students from 35 plus countries calling us their home now. Krupanidhi believes in providing quality education with training, topped by finishing school and value-added courses in addition to the regular programs. Our residential campus with multi-ethnic and diverse setting hosts more than 6,500 students who bring in uniqueness from all corners of India as well as from various geographical locations of the world. The Krupanidhi Group boasts of imparting exemplary education through several colleges under its wing, including Business School, Pharmacy College, Physiotherapy College, Degree and Pre-University College, Nursing College, and International School. KrupaPharmaCon is an annual event in the calendar of Krupanidhi College of Pharmacy, Bangalore. In this third edition of KrupaPharmaCon, we intend to showcase the emerging technologies in the pharmaceutical domain. That's why it's entitled Drug Development 4.0 Emerging Technologies. In this two-day online global conference and workshop, we intend to cover the recent advances in the technological aspect of drug development with special emphasis on emerging technologies which are increasingly deployed to rationalize and fasten drug development. I take this opportunity to welcome all our dignitaries today. Professor Dr. Suresh Nagpal, Chairman, Krupanidhi Group of Institutions. Mrs. Geeta Nagpal, Vice Chairperson, Krupanidhi Group of Institutions. Dr. Sam Paul, Director, Krupanidhi Group of Institutions. Dr. Rajendra S.V., Principal, Krupanidhi College of Pharmacy. Dr. M.D. Karvekar, Director of Academics at Krupanidhi College of Pharmacy. Professor Prakash V. Malya, Director, CPPA at Krupanidhi College of Pharmacy. And Ms. Kavita A.N., Vice Principal, Krupanidhi College of Pharmacy. I request all our dignitaries to kindly inaugurate the event by lighting of the lamp. Spreading the Almighty's grace and divine energy in the surrounding. Light also symbolizes abundance, prosperity, and knowledge. The lighting of an oil lamp is regarded as a bow down to knowledge. Thank you so much to all the dignitaries. Let us begin by calling upon the convener for the conference, Dr. Rajendra S.V., Principal, Krupanidhi College of Pharmacy. Sir has made remarkable contribution to the field of education with more than 25 years of academic experience and 15 years into research. He has been recognized with many awards and accolades in different forums and is also the lifetime member of Association of Pharmacy Teachers India and Indian Pharmacological Society. I request Sir to kindly deliver the formal welcome address.
Good evening to all of you. It gives us immense pleasure to host and witness the first virtual global conference organized by Krupanidhi College of Pharmacy, a daughter institution under the umbrella of Krupanidhi Group of Institutions, Bengaluru. I feel elated and take pride to state that this is the third edition of KrupaPharmaCon 2021 that the institution is holding virtually with the theme Drug Development 4.0 Emerging Technologies. KrupaPharmaCon 2021 is an annual event of Krupanidhi College of Pharmacy, Bengaluru, which will be a two-day virtual conference and workshop to connect guests and speakers globally and aiming to cover the recent advances in the technological aspects related to the drug development with special emphasis on emerging technologies. With this brief note, I am really honored and delighted to welcome the chief guest of today's function, Professor Dr. B. Suresh, President, Pharmacy Council of India, New Delhi, and Pro-Chancellor, JSS Academy of Higher Education and Research, Mysuru, Karnataka, India. I welcome you, sir, to this august gathering to inaugurate the two-day global conference. I feel it's my privilege to give my warmest welcome to the Krupanidhi Group of Institution leadership and the chief patron of the event, Professor Dr. Suresh Nagpal, sir, our Honorable Chairman, Krupanidhi Group of Institutions, Bengaluru. Sir, I welcome you to this two-day conference. I deem it necessary to welcome our beloved Vice Chairperson, ma'am, Mrs. Geeta Nagpal-ji, to this august gathering. Dear ma'am, you are cordially invited to this global conference. I take this opportunity to gratefully welcome our patrons of the event, Professor Sunil Samson Damangi, Secretary of Krupanidhi Group of Institutions, the Board of Directors, Mr. Akash Nagpal-ji, Mrs. Neha Nagpal-ji, Dr. Sampal Isaac, the Campus Director of our Krupanidhi Group of Institutions, and Professor Rijwan Ali Beg, the Academic Director of Krupanidhi Group of Institutions, Bengaluru. I extend my hearty welcome to our advisors of this event, the Board of Directors of Krupanidhi Group of College, Krupanidhi Group of Institutions, Professor Dr. M.D. Karmekar, Professor Prakash V. Malya, and Administrative Officer Professor Rajesh Rauri to this two-day global conference. A sincere and warmest welcome to all our eminent speakers who are joining from multiple locations globally to share their experience, expertise to the participating delegates. It's my duty to give warmest welcome to all the scientific poster evaluators to this virtual conference. It's my pleasure and duty to welcome the principals of various colleges across the country, the faculty, the young budding researchers, and the students to this global conference and workshop. I take this opportunity to sincerely welcome all our principals who are under the umbrella of Krupanidhi Group of Institutions. It gives me an immense pleasure to welcome the delegates from all over the world. It gives me an immense pleasure to welcome all our own faculty members and students to this global conference. I feel it's my bound duty to welcome all the press media persons and electronic media who have taken time to participate in this inaugural function of the 2 days global conference. Finally, on behalf of the entire team of organizers, I once again welcome all of you to this prestigious virtual platform. Thank you. Warmest welcome to all of you once again.
Thank you so much, sir. Today, we are extremely honored to have been graced by our chief guest, Professor Dr. B. Suresh, President, Pharmacy Council of India and Pro-Chancellor, JSS Academy of Higher Education and Research, Mysore, India. Sir has been, for the past 35 years, actively involved in uplifting the standards of pharmacy education and professions in India. He was elected in 2003 as the President of Pharmacy Council of India, the body regulating the pharmacy education and profession in India. He was once again re-elected as the President of the Council in August 2008 and then again in August 2013. Sir broke all records of winning the PCI presidentship for the fourth time in 2018. He was also the chairman of the scientific body of an Indian Pharmacopoeia Commission, which was responsible for setting of standards for pharmaceutical substances and formulations. Dr. Suresh is also the delegate member of the United States Pharmacopoeia Convention and also the member of the Council of Convention of the USP. He was also the chairman and member of ACPE International Services Commission USA and is the executive member of Commonwealth Pharmaceutical Association. He had pioneered the cause of promoting pharmacy practice concept in India and has the unique honor of having introduced postgraduate program in pharmacy practice and the PharmD programs in India. Dr. B. Suresh, as the founding Vice Chancellor of JSS Academy of Higher Education and Research, Mysuru, a deemed university, assumed the office in June 2008 and was nominated for a second term in 2013. He nurtured the deemed university to scale great heights in a short span of time and was successful in helping the university be ranked 34th nationally by the National Institutional Ranking Framework in 2019. MHRD Government of India ranked number 1 by the Karnataka State University Ranking Framework in 2007 under the Young University category and obtaining NAC A+ grade with a CGPA of 3.48. He broke all records of making JSS Academy of Higher Education and Research to be ranked among the top 500 universities of the world by Times Higher Education ranking, becoming the first deemed university in India to set this record. Dr. Suresh has to his credit several national and international publications and has initiated several international collaborations through bilateral, academic, and research exchange programs. He is the recipient of the FAPA Ishidate Award 2004 International Award and Doctor of Science DSc Honoris Causa conferred by the Tamil Nadu Dr. MGR Medical University, Chennai in 2007 and by the Rani Durgavati Vishwavidyalaya, Jabalpur, Madhya Pradesh in 2012. Sir, it is our immense pleasure to have you amongst us. I request you to kindly shower us with your kind words.
Institutions, Dr. Suresh Nagpal, Mrs. Geeta Nagpal, Vice Chairman, Sunil Damangi, again a good friend and a long-standing acquaintance, Principal Dr. Rajendra SV, Dr. Kavita, Vice Principal. Again, my pleasure and once again in seeing Dr. Sam Pal and Dr. Professor Rizwan on the lighting of the lamp. And of course, the 4.0 generation and leadership of Krupanidhi Group, Akash and Neha Nagpal. Very good afternoon to all of you. And at the outset, I would like to thank Professor Karvekar, who was a motivational, inspirational leader for me and a great teacher for me, who has shown me the way to pursue both professional and academic pathway for my career. It was a call from Dr. Karvekar telling me that Professor Nagpal would like to me be the chief guest of this conference and the third in the series. I recall my being there in the first and second also, so I presume that they wanted me there, and I gladly accepted, because once Dr. Karvekar says, and it is Dr. Nagpal, Suresh Nagpal's event, I cannot refuse them both. The virtual conferences that are happening now are doing both a good and a little bit of bad also. The good thing with these virtual conferences is that many of these events are taking place with a lot of eminent people coming across the world and addressing us, enlightening us with different thoughts, different advances that are happening in the field. And we don't have to even move out of the house or our offices to listen to these talks and lectures and enrich ourselves. Number two, the digitalization has reduced all the borders, and the people from anywhere in the world can participate and interact, present their scientific findings, get their certificates, present their presentations. All this is happening today in the virtual world. There are several such positives we can go on enumerating for this virtual conferences. The negatives, of course, is that we miss the opportunity of meeting in person and sharing our fellowship and the thought process which take place along with that. The second disadvantage which I feel with the virtual conferences is there are too many of these conferences happening, where perhaps the students or the teachers or people who would like to attend are finding difficult to find out which meet to attend or not. So I think now the time has come that the enhancement of quality of these virtual conference also plays a very important role, which I can visibly see how effectively technology is being used at Krupanidhi institutions, particularly this KrupaCon, which is being organized here. I can see the use of technology extensively happening there. So my congratulations to Krupanidhi group of institutions, congratulations to the leadership and faculty, and my best wishes to the students for the good organization they have joined to pursue their educational ambitions. The theme of the conference has been kept aptly as the emerging technologies that are there and the drug development 4.0. I think the basis of which has come from the pharmaceutical industry 4.0. I think it's very important to understand what is this 4.0 or 4.0, whatever we want to call, from the pharma perspective, and then look at the drug development perspective. I had just put a few slides together just to give a perspective. I do not know I can share here. I will try to attempt to share. And if so, I think the technology people can appropriately show it in their screens when they are doing it. I'll try to share my slide. Yeah, I have shared my slide, and I think it's visible to all of you, and I think my voice is also clear. So we are looking at the pharmaceutical industry when we talk of 4.0, where it will be totally technology driven. The whole industry is going to be technology driven. The human interface comes only at the stage of planning, processing, and rest of the things, leaving it to machines and the artificial intelligence and IoT to take care of the whole process of the development of pharmaceutical industry. Now, I was recently interacting with the Indian Pharmaceutical Alliance and their leadership, which is the organization of the top 25 industries, pharmaceutical industries of the country. I mean, the likes of Dr. Reddy's, Sun Pharma, Cipla, and all that big groups, 25 top 25 people, who cater to about 60% of the medicines of the country. I mean, only the remaining 35, 40,000 industries, they are there. They cater only to the 40% of the country's needs. So this big pharma, they have already put a strategy in place for moving towards Pharma 4.0. And the stepwise the process which they have put together is, initially they are going to enrich their companies to be technology driven. That means machine works in the direction of the man. That is the first phase. They have put it together. The second is the second phase they are looking at is the machine tells the man how to do it, and the man carries out certain amount of works there. The third phase they are telling is the machine would even correct the man if he's doing wrong. So it will try to tell him this is the way you are doing wrong and do it. And phase 4 they are looking at is the total lights down. That means there will be no operators inside the manufacturing facility. Everything will be automated, and there is no need for light there because all processes would be going on. They call it as the lights down stage of their industry. And this perhaps is what we are looking at the 4.0, where completely technology handles the whole manufacturing process. And with the development of medicines taking place in different facets and much faster track, I think the industry has to move on with the technology that is available. So in this direction, if you really try to understand what is this technology 4.0 or the pharma 4.0, it is totally, I think if you look at it, it is the digitalization and automation of the industry that is playing a major role in the whole manufacturing process. I think this is one of the major changes that is going to happen, would be that the digitalization and the automation would get involved now completely in the complex product portfolios and life cycles. We all know in the manufacturing of tablets or capsules or oral liquids, there are several processes, right from identifying the active ingredients, the additives that have to go with that, the granulation, punching, packaging. All these processes are very complex processes. But what they are looking at is, can digitalization and automation take care of many of these things? So it will be, in fact, a process of evolution only. The industry will slowly evolve in this direction, as I mentioned to you, in a phased manner. What they are looking at is, at the end of this 4.0, there will be a totally digitalized plant floor. That means everything will be functioning. Many of these activities which we are talking about, we have heard about the houses being connected with complete internet operated mechanisms. That is, you clap your hands, the lights will come on, and you give a voice direction, the TV get on. So these type of things we have been talking about or listening in the by people telling it, or in movies also we have seen. But this is not far away, even in the industry, where voice commands and the technical person may be watching a movie at his home or in a theater, and an alert from the plant can go to this individual through internet of things connectivity, that there is something wrong in the processing, and he can rectify wherever he is sitting using the directions that he has to give. So this is not far from reality. It is being done to many a level, but this is where the industry is moving. Now, what comes to the question is our understanding of the readiness and the maturity of the industry, as well as the people who are associated with it. That means digitalization, automation is very easily said than done. Because if you talk of digitalization, we even know, even in education, when we talked about teachers or students using digital technologies for teaching, we were reluctant in the early stages. Now, because of the COVID pandemic, we have become very confident that we can use digital technologies for our education also. The same thing is the understanding in the industry of the uses of digital technologies and how they can be valuable or useful, so that there are no errors or actions taking place. So basically, they have divided this digital maturity into 4 quadrants. The first, of course, being the resources, that is, digitalization resources are required. Then the workforce of the future, that is the pharmaceutical scientists who we are training and sending, the undergraduates and postgraduates. They should be able to understand these technologies. Just going with certain theoretical knowledge, which we try to learn in the classroom, and some practical experience of obsolete experiments that we are doing, if we go with that, I think no industry is going to find place for us. So definitely a lot of digitalization and automation issues will be discussed in the coming years in the curriculum, so that the students are familiar with this. And these human resource should become available, and they are qualified also to be competent. So it will not be far away that a B.Pharm graduate will be studying a additional course in machine learning or artificial intelligence from the application point of view and move forward while he is going for an employment. Next is the information systems, whether they are available. That means, now let us say if you are talking of integrating, I was explaining in one of the meetings that the day is not far away when the manufacturing plant need not be audited by FDA, either India or US, whichever, because the whole systems would be integrated in such a manner with the software available with the regulator that any variation or any mistake being carried out by an industry will alert the FDA about the errors being conducted by the industry, whether it is the arrival of the API into the industry or the testing of the API and its reports. All this will be so automated and connected with the basic platform that the information will keep moving further, and there's no option of doing anything wrong. It has to be there. The next is we have to develop a culture also of communication and decision making. Like when this type of decisions have to be done and steps have to be taken, everybody across the board should accept that this digitalization and automation will be a way of our life, and we have to redefine the roles of the people who are there in the manufacturing area and see how they can be best utilized. Then next is the last one is the organization and processes. That means how do we incorporate the whole life cycle management and the control strategy to be incorporated into the manufacturing. So all this thing will ultimately lead to, you have heard about quality by design. Now it will be data integrity by design. So there will be no data integrity issues coming up with the industry. I think this is what basically what we mean by Pharma 4.0. Moving further, there are 12 theses which the Pharma 4.0 is trying to talk about. I have just enumerated here. One is the operating model for medicinal products. This will be very well clearly defined. I think this is going to be the first step towards that. What will be the operating model for each of the products which we are going to look at? And that has to be defined and taken forward. Perhaps you will have what we call as continuous manufacturing processes. That means you may have one line exclusively for one product, right from API to the final packaging. So you may have to put several assemblies like that in an industry for different products you want to bring in. I think this is the where we have to understand and move forward. Now, next is to understand that this technology will be embedded with the regulations and the best practices. Let us say if GMP is a minimum requirement, the software or the technology that you are going to use cannot be compromised because GMP will be inbuilt into the system itself. And let us say if US FDA or if the Indian FDA is asking you to carry out certain tests, now unless those tests are not uploaded into the program, you will not be authorized to take anything else or any other product further for manufacturing. So it will get embedded into the system. Now next, it will serve as a bridge between the industry, regulators, healthcare professionals, and the stakeholders. That means we are going to look at an integration of the industry, the regulatory agencies. Healthcare means the physicians and other healthcare providers, the stakeholders. That means even the patients may become a part of the whole process of understanding the 4.0 industry which we are talking about. Now last is, this is what I would like to say is, this will be the basic enabler and a business case when the industry is going to talk in future to the in the market. They are not going to talk to them that this drug is the best. They are going to tell how this drug has been made best for them. So I think that will be the business case. When they talk to the patient, why should they take a particular product? They would be able to explain them that such rigorous controls have moved forward. Then it will have be established, this for the established products, new business cases would have to be developed. That means already some products are in market. How do we move forward with that? And innovative approaches for business cases calculation should be move forward. All these investments that will go into them may become the cost of the medicine also in the process, and new idea of looking at. Now particularly we all know we go at the product cost, that means the APIs and other materials went into the cost plus the markup for the industry, and then we are looking at the availability at the market with the suppliers, that means the wholesaler and retailer, his service charges all put together. We look at the pricing. Now technology also will become a part of the thing. Then no longer it will be by a choice. It will become that these industries will have to go for PQS, that is pre-qualification. Support should be available, and the controlled process and products would be also involved in this. That means it will become a prerequisite for 4.0 industry. If you want to define yourself, I think this will be the next level. We should not understand it is totally an IT project. That means only internet and communication tools are going to play a role. No, it is beyond that. There are also organizational, cultural processes, resource aspects. All this will be incorporated to 4.0. And the maturity model will allow the industries to align and operate innovative, and also supply. It will also link the established industries, suppliers, contractors to an appropriate level which it will be required. It will be integrating that. It will become a competitive advantage for an industry. It is, will you ask that industry 4.0 will be applicable to all the industries, whether it will become a law? No, it may not become a law, but it will provide a competitive advantage to the person. From what the industries look at today, that is from blockbuster drugs to niche products and personalized medicines, 4.0 would be the way forward. Let us say today we have one company has one blockbuster drug, and it will keep on moving with that. The COVID pandemic has shown that any drug is capable of producing the beneficial effect which we are looking at, and be able to take it further for our use. Now, moving further, I am just trying to show the first slide. I showed you the inner box, that how this digital maturity will take place. Now we are going to look besides the maturity, the impact also. So what will happen is it will become a plug and produce type of facility. That means you get into a particular facility, all the system is there, and all you have to do is define what product you want to do, and it will take. Then it will also build the management and communication strategy would be there. Then there will be what you call as process automation and process maps, and critical thinking will become a part of it. Then there will be a holistic control strategy will exist, and finally the validation of 4.0 would take place. And this is where the industry is moving in this direction. Further, now what will happen with the digital transformation and the technology which is going to come, which is going to be linked with digitization, automation. The earlier you look at it, the process was manufacturing was raw starting material, then bioproduction, that is you produce the product, and then purification and packaging and move forward. This particularly we have taken the example of biological drugs. Now we have to go backwards also, look at the supply chain, look at the data information which may be available, logistics that will be associated, then talk about raw material starting and other things. And then finally, the maintenance part of it also will become a part. So the whole gamut of digitalization and digital transformation is going to develop backwards and forwards in advanced level is what we am trying to emphasize. Now, just I gave you a sample of how a manufacturing facility would get evolved. Now, what I am trying to tell is next level now is how a product is also going to be a different from such facilities. Now, you may be aware that in 2017, the US FDA has approved the first smart pill. That means in this particular drug, that is aripiprazole tablets, it is having a sensor along with that. This tablet has a sensor with it. So as the patient takes the pill, it will go into his body, and he has a patch attached to his body. So the tablet, once it goes on, it will send a signal to the patch. And from the patch, the information will go to the smartphone, and from there it will go to the physician or his relative or whoever he wants to know the information that I have taken the tablet, and the healthcare provider tracks the adherence over the time. Did this person take it? Particularly, it becomes very valuable in particularly say schizophrenia and certain mental disorders and other places. We don't know whether the person has taken the tablet on time or not, and it may lead to other type of challenges that may come in. This is just a simple example of this thing. Similar technologies are being explored for maintaining blood sugar levels, controlling hypertension, using in Parkinsonism, using in Alzheimer's, all these type of diseases where chronic type of impact is required, and medication becomes a mandatory requirement to control and maintain the things. I think this type of new technologies will be moving forward. So I think this again a sample to you just to give the flavor of 4.0. And similarly, when we are talking of these technologies, we as the education providers or institutions should be ready to meet at these type of things, and accordingly modify our approach towards that. Now, coming to the drug development and the drug discovery phase itself, we all know how the whole process goes through. That is, we have nearly 5,000 to 20,000 compounds to begin with. That is the drug research, drug discovery path. We try to screen it. From that, we take about 250 chemicals or compounds to the preclinical stage. From that, 5 end up in the clinical stage, and then 1 comes to the approved drug stage. Like you start off with 20,000 products, and over a period of 10 years, we get 1 product after spending $1 billion. This is what we have been learning and doing for. I think you all are aware that how in the shortest possible time, the Pfizer came out with its vaccine. In 6 months' time, the vaccine was available by collaborating, putting large number of teams together, using different types of digital and technological platforms to achieve its outcomes. And it was all possible because the whole organization worked with one focus and made sure that it is achieved. So this is a standard traditional drug discovery development which you go. Now what has come is further, we have got now digital tools that are available to us. Now, you know, there are several such digital tools available to you. One is now, for example, you have the Schrödinger's drug discovery platform is available to you, where you have got billions of compounds already synthesized and it is available. The receptor sites are available. Now, all you have to do is use this software and platform and start your drug discovery development. And with that type of thing, when you go on looking at what type of drug you need, finally you narrow down to 4 or 5 compounds which actually go for synthesis, and then it goes to the thing. Same thing, they are having a platform even to decide on the dissolution kinetics, what type of solvent it will be better for using that, what will be the absorb PK/PD studies can be done using, can be determined without the industry spending a lot of time on PK/PD efforts to go through. There's such like a sure fit solution comes in. Again here, when this type of machine learning addition comes, there is a multiple parameter optimization in silico that is available here with the software. Then the development of candidate takes place. Then you do the synthesis and assay. Then you carry out the structure and analysis, and you have the identification of the compounds which you achieved, and thereby you again use a structural biology to achieve the outcomes. So these type of platforms are already available to you, and you have to only learn to use them. Again, I would like to say here, the Pharmacy Council of India did an effort to make available these platforms to the institutions in the drug discovery hackathon. We got it 3 months free of cost for all the institutions, but there are very few takers who are interested to move forward. Now, if we are satisfied with knowing our own regular chemistry and being able to teach that and finish our teaching assignments, neither the institution would be able to progress further in the research and development where the industry is moving, nor our students would be able to find opportunities if they are not able to comprehend the advances that are happening in the field. So it is very important that the pharmacy education also moves in the same level and speed. It will cost the institutions, but definitely this cost is very important, because with this cost only your student is going to be enriched. And if the student is not enriched, in a few years' time, we wouldn't have takers of these students. So it's very important for the students to move forward and be able to successfully be available. Even the institutions move to the 4.0 model, and that's where I come that pharmacy education 4.0 is already here. We have seen the fringes of digital use for the purpose of teaching purposes. Now we have to extend this further to the what you call as to our research and technological understanding in the industry and all other aspects through augmented reality and virtual reality. We'll have to move forward and interface with the industry and really bring the technology. Can a live manufacturing process be telecasted into the classroom? I think these are the ways we have to look for the future, and that is where we are leading to you. And I once again congratulate the Krupa group of institutions, Krupanidhi group of institutions, for the wonderful work they are doing, the level of quality they are maintaining at the institution, and the best efforts put by the faculty and the leadership there. My congratulations to the Krupa group of institutions, Krupanidhi group of institutions, and my special thanks and respects to my teacher and mentor, Dr. Karvekar. I thank you all for inviting me for this meeting. I got great pleasure in inaugurating and wish this 3-day conference all the success. Thank you very much.
Thank you so much, sir, for those words of wisdom that are as knowledgeable as ever. As we move ahead, it is my immense pleasure to invite the lady who is an embodiment of empathy and charm, Mrs. Geeta Nagpal, Vice Chairperson, Krupanidhi Group of Institutions, the lady whose idea has given shape to the very institute that we stand on today. She is an educationalist and a philanthropist who is also a strong supporter of performing arts and women entrepreneurship. She is a lady with business acumen as high as her emotional quotient and often offers free counseling to the ones in need. Ma'am has been the recipient of several awards and accolades and has spoken at diverse forums advocating quality in education. Recently, she was also felicitated with Women Empowerment Principles Leadership Awards 2021 by Asia One magazine. It is an honor to have you with us, ma'am. We welcome you and request you to kindly shower us with your kind words.
Before I say anything, I must confess that I've almost forgotten how to address a forum because of long break, lockdown, stay home, stay safe orders, continuously happening from long time. But it's a kind of a rehearsal for me today. But having said that, I understand your appreciation and magnanimous attitude of accepting it. Thank you. Talking of today's event, before that, I would like to say I welcome everyone with my greetings and warm salutations. Today's event, indeed, it's a great pleasure and a privilege to say a few words on this platform. We have with us very eminent speakers, and also they are a galaxy of stars who are from the pharmacy field. And also we have enthusiastic and a very brilliant audiences who are stars in the making. As far as topic of today's is concerned, it's about emerging technology in the field of pharmacy. the drug development, etc. When it is emerging technology, what strikes me is about the artificial intelligence and machine learning, which is there in every field. Having said that, I would like to understand the speaker's perspective, how they are going to apply this in the drug development case, and also would like to know if there is any progress it gives rise to the better quality of drugs or anything as such. I sincerely understand the contribution of the pharmacist, because this pharmacy is existing from very, very ancient times. And I know that your education, your knowledge, and your research is helping lots and lots of people because of the medications, what you give it to the humanity and to the society. We are all having one or the other kind of illnesses because of the stress, because of the eating habits, because of so many things. But everything is a quick fix in the pharmacy. I just popped a pill in the morning for allergy, that is a cetirizine. And I understand you all are doing a great service to the humanity with your entire learning and service to the humanity. Thank you so much. And I appeal to all the delegates to enjoy the conference in such a way that eat the conference, walk the conference, and talk the conference. Think of the certificate, but not too much. Thank you.
Thank you so much, ma'am. It was delightful to listen to you as always. Krupanidhi Group of Institutions started more than 3 decades ago. It was and is the product of the vision held by a man who dreamt of making top-in-class education possible and accessible to every desirous one. The man with the vision is our founder, chairman, Professor Dr. Suresh Nagpal, who is an educationist and a visionary who, with over 3 decades of efforts, has brought about a change of a lifetime in the education system. Sir has revolutionized the way education is imparted through the many colleges under Krupanidhi Group of Institutions wings. His efforts for changing the way education has been looked at has been recognized with numerous awards. Sir has been awarded the Academic Visionary Award for Excellence in Education, Leadership and Innovation in the Academicians category of the WCRC Leaders Asian Education Excellence Summit and Awards. Today, Sir has left no stone unturned in developing world-class education system at Krupanidhi that houses students from over 35 countries. We are more than honored to have you with us, sir. Up next, I would like to invite the person who is like the wind beneath the Krupanidhi wings, Dr. Sam Paul Isaac, Director, Krupanidhi Group of Institutions. With over 20 years of experience into the field of education, Sir has worked in diverse capacities towards bringing about upliftment in the education quality and system. As an efficient administrator and an industrious person with multiple talents, Sir has several laurels and awards to his credit. His vision is to inculcate research and innovation into all educational streams, starting from the school education to post-graduation programs. We are glad to have you amongst us today, sir. A warm welcome and request you to share some words with us.
Good afternoon to one and all. It's indeed my pleasure to be a part of this wonderful conference, entitled Emerging Technologies in Drug Development 4.0. What an amazing conference that we are having. When we talk about technology, we couldn't think about technology that, we are going to sit virtually for a conference. If we talk about it about 3 years back, nobody would ever dream that we would have a conference where we are, addressing thousands of people sitting in the comfort of their home. And that is possible because of technology, right? So each of you all are sitting wherever your place you're sitting, but you're still able to be a part of the conference. That's amazing. The technology, there's a big revolution that's taking place, and there's a great disruption that's happening. one of the Greek philosopher said, change is only constant, which is very much relevant to the pharmaceutical field, because without change, the critical decisions of the healthcare cannot be taken. There is a constant change that happens. The technology that we are right now in, is something different, where the drug development and discovery is happening at a faster rate than ever we could imagine, because of the artificial intelligence, machine learning, as ma'am was telling about the artificial intelligence and machine learning and data analytics. And because of these things, we have seen the things that are developing very fast. Now, when we talk about the vaccines that have come so fast, it is not possible without having the technology. Why this kind of a conference is very important for students and faculty is we need to update ourselves. What we learn in academic institute is basically becomes a disconnect when you are in an industry, right? Industry is going at a faster pace in technology, whereas in academic we see the curriculum going in a different phase. There's a different mismatch. These kind of conferences would definitely fill those gaps, and the students or the faculty who are here, you'll understand what are the technologies that are coming up and what are the new things coming up. You need to bridge yourself. The latest thing that we're looking at, where we collaborate, the academia and industry, is very, very important. And we have today eminent speakers from the industry as well as an academic. I think if the best of their knowledge, I think they can impart the great knowledge and wisdom to the students and the faculty we are part of this conference. And I'm sure at the end of this conference for 2 days, you are going to carry something home. I believe it's not just going to be a webinar or a conference that happens in a normal way, but this is something that you're going to carry something home and going to make a difference in your career, and that is very important. I urge you all to just be a part and encourage, and thanks for thousands of entries that have come in and being a participant here, and that's just amazing and wonderful. Once again, thank you everyone. Welcome to KrupaPharmaCon 2001. And I urge you all to be a part of this 2 days. Don't log out, be logged in, and you're going to see great things happening. Thank you so much.
Thank you so much, sir, for the quick food for thought that your talk often provides us with. And now, welcoming another dignitary. Krupanidhi believes in holistic development of its student, and the man who brought this concept to life is Professor Prakash V. Malya, Director of Center for Pharmaceutical Professional Advancement, also known as CPPA, at Krupanidhi College of Pharmacy. Sir has traveled to more than 70 countries and is backed by experience of more than 3 decades in corporate, in numerous pharmaceutical industries at different capacities like global regulatory affairs, international sales and marketing, product management, ministry liaison, new product launch at national and at global level. Sir chose to be the interface between academics and industry at Krupanidhi. He has passion for teaching, training, and student development, and uses the experience gathered to impart knowledge and aid in development of the students. It is our pleasure to welcome you, sir. Request you to kindly share your words of wisdom with us all.
Thank you for the nice introduction, and my very happy to be amongst an august group of dignitaries on and off the dais. And it is a historic conference, first of its kind from Krupanidhi, and I'm very much pleased to be here on the stage when such an event has taken place. Right from the planning stage till the stage we are about to launch, that is today, the D-Day as we call it, we have done in record time. thanks to the coordinated and team effort by the management who supported us wholeheartedly, the directors, our principal, vice principal, the other staff, directors, and all of us together. In the last few days, the emphasis was on speed, on accuracy, and efficiency. And all the three came together. And here we are today. The conference is about to start. Like my previous speakers who complimented that there is a galaxy of speakers towards the 2 days deliberation, I might add one thing, that speakers are from all over the world, not just from our country. And all of them have complimented one aspect about the theme. And what is that theme? Drug development. Not only that, we didn't stop there, but there is a digital figure, the alphanumeric part of that. The numeric part is 4.0. 4.0. That is the numeric figure on which there is a lot of emphasis. So everybody was wondering, what is 4.0? So drug development 4.0 and emerging technologies. I want to add some few words on what this 4.0 means. 4.0 stands for Industrial Revolution. Okay, that started way back in the 17th century. Let me briefly introduce what happened in each of those centuries from 1.0. When it started as 1.0 in the mid-1700s. From mid-1700s to mid-1800s, that is called as the mechanical revolution. In that, all kinds of handwork shifted to machine work. So a revolution takes place, and a lot of people who are doing hand type of jobs, like cobbler, like goldsmith, the iron master, all of them had to shift to the mechanical part of the revolution. As a result, we had machines, we had inventions in agriculture, we had invention in other endeavors of machinery, and so ended the first 0.0, that is 1.0. Then the second, that is the second revolution, the second industrial revolution, started as 2.0 from mid-1800 to mid-1900. This is called as the technological revolution, when we had the railroads, we had the electricity, which had come in, and these were the mainstay of, endeavor of that era. Then the third revolution, that is 3.0, started from mid-90s to 2010 or 2015. And that revolution is called as a digital revolution. So we started from mechanical revolution to technological revolution to digital revolution. And now comes 4.0, where we all are now in the present generation. These are the emerging technologies, the revolution of emerging technologies. Slowly from the computing era of the third revolution till the communication technological era of 3.0, we have now come to 4.0. And what is there in 4.0 is the total emerging technologies. World has moved very fast. Now we are talking about artificial intelligence. We are talking about machine learning. We are talking of Internet of Things. we are talking about robotics. And in the pharma field, we are talking about genomics. We are talking about personalized medicine. And it keeps on going. We have AR, VR, and MR. That is augmented realities, virtual realities, and mixed realities. And there's another special emerging technology, which is called as blockchain. So all these things are merging together and forming the 4.0. So in the 2 days deliberation, what everybody will be happy to note is that everybody, first of all, appreciated the title itself, the theme itself, and they also appreciated the fact that it is so broad-based and it is so pharma-centric, because pharma domain is in the center of a whole lot of revolution. We heard about it during this pandemic. And why? Because there was so much ado about vaccination. And who were the pioneers in vaccination? Normally, a 10-year period for vaccination from the start to finish took only 10 months during the pandemic. And that is something which has to be applauded to all the scientists in the pharma and the other domains, medical domain and the pharma domain all over the world. They came together. A 10-year-long period from discovery to marketing has just taken 10 months. And today we stand here proudly with having both our jabs here have taken as vaccination, and we feel that we are all, safe. Of course, we have to take all the precautions, but the factor is there. So the 4.0 revolution signifies so many things. And those who are going to speak today in the later part and tomorrow will be talking about various subjects. There are no two themes or no two topics repeated. And that is the beauty of this KrupaCon. There'll be topics on, on COV, SARS-2, and there'll be topic on cancer research. There'll be topic on genomics. There'll be topic on monoclonal antibodies. There'll be topic on biosimilars, and so on and so forth. Not only that, we have a hands-on workshop on the second day, and above all, we also have an excited and a very heated type of a discussion during the, I hope so, because 6 people are there in a panel discussion. Besides that, our staff have made exemplary, efforts to see that a lot of poster presentations are there in a different hall altogether. You can see, when you see the layout of the map, you see that this is on a different hall. And a lot of students have enthusiastically participated, and they have already registered themselves. And we have a huge amount of registration this time, despite of so many other conferences going on at different levels in the country. But ours has been the highest number of people who have registered. And thanks to our senior people in the colleges, like the principal, vice principal, and the entire staff team. I am sure that these 2 days will be beneficial to the academia, to the industry, to the faculty, and even to above all, our student friends. So take maximum advantage of the 2 days conference. And my theme, the ending theme of this is KEY, which means keep educating yourself. And this conference will give you a platform to educate yourself. I wish the conference all the success. I thank all my, team members, the team members of Krupanidhi College of Pharmacy, as well as Krupanidhi Group of Institutions, and above all, the management. And I wish you all the best for this deliberation. Thank you very much. Thank you for inviting me.
Thank you so much, sir. Who best to explain how big a industry pharmacy field is, and to show the link that, of course, drug development has reached its 4.0. At Krupanidhi, we have been blessed to receive guidance from the teacher who has been substantial in shaping many of the teachers here at present. Dr. M.D. Karvekar, Director of Academics at Krupanidhi College of Pharmacy. Sir, with over 45 years of experience in the field of academia, administration, and regulatory, has made remarkable contributions to the field of pharmacy education. Sir has served in various capacities over the years, like Chairman of Board of Studies in RGUHS and numerous other universities, Member Secretary of Board of Examining Authority, Government of Karnataka, Executive Member, Pharmacy Council of India, Member of Drug Technical Advisory Board, to name a few. It is an honor, sir, to have you amongst us, and it would only be appropriate to receive benediction from the teacher of teachers himself. We request you, sir, to kindly take over the floor and shower your blessings upon us.
Good evening, dear dignitaries who are all present over in this prestigious virtual platform for the inaugural function of the KrupaPharmaCon 2021. I feel it is an honor to be a part of this program and addressing the global conference themed Drug Development 4.0 Emerging Technologies. I am proud to announce that the global conference is hosted by Krupanidhi College of Pharmacy, Bangalore, which is a leading educator in the pharmaceutical and healthcare space in India. The institution offers diploma to doctoral level programs in the domain of pharmaceutical sciences with very high acclaim. As an institution with close collaboration with industry, Krupanidhi College of Pharmacy is organizing this annual conference, KrupaPharmaCon 2021, which provides a collaborative learning platform for the luminaries from industry, academia, and regulatory stakeholders. I am really elated to witness this global conference being organized despite prevailing COVID situation in this third edition of KrupaPharmaCon 2021. The conference intends to showcase the emerging technologies in the pharmaceutical domain titled the Drug Development 4.0 Emerging Technologies. The importance of this is already highlighted by one of our friends. The theme is very innovative, and the participant will be given a holistic learning and exposure to technologies in the drug development and discovery, and its leverage in the drug development. Also, this conference creates a forum for the participants to appreciate the use of emerging technologies in the drug development and also showcase their work in the emerging areas. I should say the participants that the local organizing committee organized this program flawlessly and in a very, very superb manner, and they should be deeply appreciated. I believe that the conference will be successful as the topic chosen are highly advanced and informative. And I was given information that quite a good number of scientific research papers are going to be presented, and for the success, what else is required. I should add that this is always a pleasure to participate in a productive knowledge, stimulating and well-run conference. I am sure that all the participating faculty, the PhD scholars, and budding researchers will surely benefit from this eminent speakers of the national and international repute. I always wish a huge success to the ongoing conference and my best wishes to all of you. I wish you a good luck to all of you. Thank you very much.
Thank you so much, sir. And I thank all the dignitaries for being part of the inaugural function today. Now, we officially start all the scientific sessions lined up for day one at Athenaeum, the main hall. We have 3 speakers today. The first session will be by Dr. Harsha Dodihal, Senior Director at Glenmark Pharmaceuticals, Senior Oncologist at Fortis Cancer Institute. His topic is Technology and Targets Reshaping Cancer Treatment. Our second speaker for the day is Dr. Onkar Prakash Kulkarni, who is Associate Professor at Department of Pharmacy, Birla Institute of Technology and Science, Pilani, Hyderabad. His topic is Novel Therapeutic Targets for Renal Diseases. Our third speaker for the day is Dr. G. Jagadish, Senior Scientist, Rockville, Maryland, USA. His topic is Development of Potential Repurposable Drugs for COVID-19 based on the life cycle of SARS-CoV-2. I hope all our delegates have a riveting exchange of knowledge and information these 2 days, and we officially start with the first session of the day. But before that, let's hear it from Mr. Rajendran R., CEO and founder of GreenChem, who sends his best wishes for this 3rd edition of KrupaPharmaCon 2021.
Dear Dr. Suresh Nagpal, good day to you and your team. Today is a wonderful day, and you have taken up a big task of starting the KrupaPharma 2021, the 3rd global online conference and workshop on drug development and emerging technologies in pharma area. And it's not an easy subject. It's a very tough subject because there are new products, there are new concepts, there are new regulatory rules. There are so many have come in the food industry and the health industry, and it's both in pharma and health industry is going to be a big chapter beginning to know more and more. And I'm sure that your students, all the students all over the world, they'll get the benefit, and then they will apply their mind to do some lot of research. And if the students or the participants have any doubt, they can contact you, and we can also assist you in making the reply, and we can all grow together. Thank you so much, and wish you all the best.
Moving ahead, before we start officially, there are a few glimpses because we couldn't take you to our college, or rather invite you to our college this year, which we would have loved to do like all the other years, give you your experience or feel of the college. We thought, why not give you a virtual feel of the college as well, along with the conference. So next few minutes, just look at how our college looks for the ones who might have not been here before. Also for these students who haven't been in college for quite some time, let's go back down time. Let's look at the college for some time. And then we start off with all of our sessions. Thank you, everyone. I hope you have amazing first day, and I hope to see you all tomorrow as well. Thank you all.
I'd like to introduce our first speaker for the day, Dr. Harsha Dodihal, Senior Director at Glenmark Pharmaceuticals and Senior Oncologist at Fortis Cancer Institute. His topic is Technology and Targets Reshaping Cancer Treatment. Dr. Harsha is a Senior Director at Glenmark Pharmaceuticals and a Senior Oncologist at Fortis Cancer Institute. He has 20 years of clinical experience and industry experience of 11 years. He has been involved in oncology drug development, which includes monoclonal antibodies, bispecific antibodies, and small molecules. He is co-founder and board member of Prana Enablers Private Limited. It is our pleasure, Dr. Harsha, to have you with us. The floor is all yours.
Everyone, just give me a second till I share my. Is the screen visible now?
Yes, sir.
Yes, thank you. At the first instance, I would like to thank the Krupanidhi group of institutions, Dr. Nagpal sir, Dr. Malya sir, Dr. Pavekar sir, and the other dignitaries for providing me with this opportunity to participate in this wonderful virtual conference, one of its kind. The last 10-15 minutes, I was virtually taken to the campus, and I thought that the bonhomie, the celebrations, the song and the dance probably was far more interesting. And considering that at 3 p.m. normally is the siesta time, and it would take me an effort to kind of hold the attention. The pharmaceutical industry, as well as healthcare per se, in the last 20 years has seen tremendous changes. And being an oncologist, I have been privy to utilize the advancements made from multiple aspects into my practice. And as a person who is involved with the industry, as a clinical researcher, as a drug development guy, it is fascinating to see how the discoveries and the technologies are emerging, which kind of give me hope that in the times to come, we will have even better therapeutics at hand. With that, I would like to jump into my presentation. If you're seeing this colorful photograph, which is, and wondering what is it all about, too many colors, interesting patterns. This is nothing but inside of a cell. This is a beautiful illustration thanks to a company called as Design. And again, this has not come through one single day. This is the actual visualization helped by multiple technologies like X-ray, NMR, and cryo-electron microscopy. These are the technological advancements which has kind of made it easier to visualize a cell in its minute detail. So you're able to kind of look into the cell, look into the different pathways, what is at the surface, and how they interplay. So just to illustrate a few which are close to me, and so these terminologies may not be very relevant to each and every person in the audience, but as an oncologist, when we hear c-KIT, when we hear EGFR, and to see this EGFR across the spectrum, like the way we are able to see it, is fascinating indeed. The AKT, then there's something called as MEK, and just to highlight the cell membrane. I just highlighted few of them from a perspective that this is the granularity at which we are able to kind of look at a particular cell and the pathways which are involved in it. In a very ideal scenario, I would have loved to ask questions and be amongst the audience, but then considering these are different times, I would proceed ahead. And since we have labeled it as technology, just to highlight, okay, what is the big deal that we have been able to see the cell, the different receptors, the different pathways, unless and until I'm able to practically apply it in terms of helping the human race or in general, the application of this technology, the application of the science, the application of the basic research, That's what we call it as technology. So just taking the help of that particular definition. If you're aware of this, these are different locks, and this comes from somewhere in Europe, where people who are in love go and tie around this locks across this bridge. The idea is not to elucidate that part. The idea is to say that as you saw that cell and the different receptors which were there, over there, each one of them is like a lock. And if you see here, there are multiple locks here. And why do we require those many locks or those many receptors on a particular cell? Each one has its own function. And sometimes the functions overlap. There are redundancies in place. What I mean by that, let us say the cell has to multiply, and at those times, one particular lock has to get opened. The cell has to produce insulin, another lock has to get opened. The cell has to produce, let us say, thyroxine, the other lock has to get opened. So the keys are the different proteins which come, whether we call them cytokines, hormones, whatever, paracrines, and they bind to the different locks and then they open up. This is a simplification of the previous cell image as well as what I'm going to speak in the next couple of slides. And what happens in cancer is that sometimes one of the lock either gets mutated or the key which comes and sits on the lock has become numerous in nature. So let us say it is a red one, which is the one which is become mutated in nature, which makes the cell grow more. Now, how am I going to identify in the first place amongst all the logs that this red one is the culprit? What are the tools available to me? I think these are things which you probably kind of read through and I'll be just touching upon. So having the knowledge and then applying the technology to kind of identify which particular pathway or which particular lock is the one which is wrong is again an interesting aspect. This is just to highlight that the way the different locks were there, you will see that whether we can start from a particular disease. We know that there's a malaria, and then we can go and find how malaria needs to be treated. What are the different ways in which you can treat malaria? Trying to produce a vaccine for either malaria or dengue has been a holy grail since many, many years. Otherwise, we have the other way around, that we understand the cell today. We know multiple targets, and we stop the functioning of a particular target. Then we understand that, oh, that leads to a particular pathology. So from cause to effect or effect to cause, the studies can be numerous. And the illustration is that the shape of the key, if it is slightly different, it may not fit into the particular lock. And then the desired effect may not be there. And in all practical purposes inside the human body, the amount of key required would also be important. That is, only on a threshold above a particular concentration, the key will be able to kind of make the necessary changes across the lock or block the lock completely. So from identifying how a cell is there, what an organ has a pathology, and then identifying the targets and finding the respective keys is a long journey overall. Dr. B. Suresh touched upon multiple aspects of what Industry 4.0 means and how artificial intelligence, other advancements are happening. These are some actual photographs taken from application. And if you look at it, one of the things which is showing across is that there are thousands, millions of compounds which are stored. And out of millions of compounds, so the millions of compounds were the keys which I was mentioning. How do I kind of identify them? And today, in an automated robotic system, high throughput or medium throughput screening can be done, and thousands of samples can be screened every day. That is, you're able to screen thousands of samples in one single day. And then there are flexible workstations with multiple arms where compound preparing and testing can be done. At the same time, the third one, images of a prototype of a novel miniaturized design synthesis test analyze facility for automated drug discovery. So the right-hand side is from AstraZeneca, while as the other one is from Boehringer Ingelheim Pharma. So the pharma has already adopted technologies wherein we are using the computational abilities, digitization, and other automated things to kind of screen these thousands of molecules, prepare them in a faster manner, and then test and deploy them. So this difficult made easy, in sense if I have to delve deeper into from the oncological perspective, it has long been held a belief that inside the cell membrane, so as you go inside the cell membrane, there is cytoplasm and then there's a nucleoplasm. What is in the nucleoplasm or what is inside the nucleus is difficult to kind of target. Kinases have been known to, KRAS especially, a difficult target to target. But now in the last 5-7 years, we have molecules which can target KRAS too. So what I have illustrated in this particular table is that from X-ray crystallography, one of the methods through which you can do structure-based design and broad kinase panel profiling is available. Sensitive biophysical methods like NMR, SPR, that is MRI concept-based, high-performance ability-based bead screening, and tethering of covalent. So these different kinds of technologies have been practically applied in the last few years to produce molecules which I'm using as of today in my day-to-day practice. So this is what I meant from that cell image, which was far more colorful and exciting. If I have to simplify and say, this is the cell membrane, and then there's a VEGFR, then there is a c-MET, then there's a HER2, then there's an insulin-like growth factor. HER2 is a kind of epidermal growth factor, EGF, the hepatocyte growth factor, then vascular endothelial growth factor. So these are all different kinds of receptors which are present on the cell membrane, which have different functions. So to simplify it and not to make you to put to sleep, if I had to say cell cycle progression, proliferation, differentiation, invasion and survival, protein translation, these are the different kinds of the functions of the particular pathways. And these pathways become rogue when a cell becomes cancerous in nature. How does a cell become cancerous? What are the reasons why it becomes cancerous? That is not of the interest today. As an oncologist, normally I would like to speak about it regarding how viruses can influence cancer, how cigarette smoking, how alcohol, etc. But suffices to say that one or the other pathway or one or the other tumor suppressor gene has to get mutated for a particular cell to become cancerous in nature, and it does not happen in one particular day. It is over a period of time. Having identified this, having known the hallmarks of cancer, we have developed different kinds of keys. So if I look at it to target the RAS pathways, we have a lapatinib which has been approved in breast cancer. We have tipifarnib and lomofarnib, which is the pathway to be approved. Then you have sorafenib, which is approved in RCC, that is renal cell carcinoma, hepatocellular carcinoma. You have got bevacizumab, which is approved in lung cancer. Pathways and drugs, if I have to name from the perspective that the trastuzumab, which is in the HER2, which was one of the first targeted therapies to be kind of discovered, which I'll come to in another slide, and then the BCR-ABL for the chronic myeloid leukemia. The reason to kind of put this across is to also bring about the highlight that when you block one pathway, it is not necessarily that it's successful. When you block a particular pathway, let us say EGFR, there could be a possibility that the downward pathways get upregulated. What I mean by that is that they get upregulated through various other signals and a resistance to a particular molecule can occur. And how do we identify this resistance? Again, we take the help of the technology. We take the biopsy samples from the patients who have progressed on a particular molecule and then go and understand the genetic as well as the proteomic part of it. And then we know that in a patient who has been taking, let us say, erlotinib for a long period of time, there is something called as T790M mutation. And then once you identify that there's a T790M mutation, you bring in a new drug called as osimertinib. So the understanding has not come over in a single day. It's over a period of time that different pathways have been elucidated, different resistance mechanisms have been elucidated. And today we have got multiple molecules which can attack through multiple pathways. That means multiple keys have been found for the different locks which I mentioned previously. If any questions are there and you want to ask it through, please put it across in the Q&A or chat box. I'll be happy to stop and then take your questions. Moving on, how does this locks or how do these keys get created in the first place? Okay, I'll pause here for you to take a look at this particular image. I hope it is playing on your screens too. And what I'm highlighting, since I could not bring in the DNA out here, but what you see over here is nothing but the RNA, which is the mRNA. And what you see which is rotating is the translational RNA. And what you see small beads above top is the amino acids which is being synthesized. So it is nothing but the way the proteins get synthesized in our body. So whether the EGFR, which I showed, whether it is the antibody, whether it is an antigen, they are all mostly proteins. Of course, glycosans are also there. And these are synthesized at the command of the DNA. The DNA has the master structure or the master key, which is able to kind of educate the particular cell to produce a particular protein, a particular thing. Now, why is this of significance? In sense, the era of genomics, if you understand how a particular DNA area is damaged or how a particular mRNA can be changed, you can probably change the way these proteins are produced. And today, we did hear multiple speakers, including Dr. B. Suresh, speaking about the fact that the COVID or the coronavirus vaccine came about in 6 months, which is not the complete truth. And I will be probably highlighting why it is not a complete truth that it came in 6 months or 10 months period. But if you look at this, right now the image, it is nothing but the mRNA pathway which is being created. So you took the particular RNA from the coronavirus, inserted into, for instance, covered it with a lipid layer and put it as a vaccine to the body. And then it goes inside our body. It is kind of able to create that protein. Once our antibodies, that is the army inside our body, is able to identify the spike protein, it creates the further antibodies. And therefore, if at all a coronavirus enters our body, there is protection against it because there are antibodies. So the mRNA technology which was there has been utilized beautifully to create the Pfizer's vaccine, which is with the BioNTech, as well as the vaccine by Moderna. But the real story comes here. The 1960 was the first time when mRNA was kind of discovered. And for the last 40 years, a lot of research has been happening in terms of identifying how we could use this technology to create an effective vaccine against cancer. Why cancer? Again, let us say if you know that a particular cancer has a particular antigen, and this antigen is created, as I showed in the previously, by a piece of RNA. So I take that particular RNA, cover it with a lipid, and insert it as a vaccine inside a human body. Then, if our T cells and the B cells have been primed to that, that means they have created antibody as well as the memory T cells have the memory towards the particular antigen. Whenever a cancer cell is surfacing with that particular antigenic property, the army inside us, which is T cells and B cells, will be able to easily go inside, attack them, and eliminate the cancer. But unfortunately, we have not had a clinical success with the mRNA-based vaccine for cancer. And there are multiple reasons for it, which I will not touch upon, but melanoma, as long back as even in 2008, 2009, these vaccines have been tried in human trials, but so far we have not had great successes. We have had one dendritic cell vaccine which was available, Provenge, which was for prostate cancer, but none based on the mRNA technology. Both Moderna as well as BioNTech, the Pfizer which partnered with, were trying their efforts and still continued their efforts of trying to develop a vaccine against cancer. And since that technology and the platform was readily available, the moment Corona came in, the moment COVID started, this platform was used to create the vaccine against COVID. And that's the reason I said that it was not 8 months. Of course, it was a tremendous development. The timelines for the development of molecules has shortened, which again I'll be depicting in another slide. And it is indeed a commendable job that the whole world came together with the collaboration of multiple scientists and the population to successfully create this vaccine as well as test it in a phase 1, phase 2 trials and bring it to the general public. Taking again a pause, and since we did discuss about the mRNA, I have already titled it here, so you probably know what is this all about. CRISPR is again a very fascinating technology which is emerging in nature. What do you mean by instance? Now we know that if a particular protein has to be synthesized, if something has to happen in our body, the DNA has to give instruction to the RNA, and the RNA has to create through the ribosomes the particular proteins or the particular molecule, antibody. Now, a segment of the DNA, if you are able to cut, you know that that particular segment of DNA is damaged or is not functional properly. And if you have a scissors to cut them, which we call it as nucleosomes, and then replace it with a DNA of your RNA of your choice, go ahead and stick it inside. That is what is CRISPR technology all about. And it provides fascinating possibilities today. As of today, we do not have anything in clinic which is backed up by CRISPR technology. But this technology is not just restricted to the creation of drug molecules. It could be used in diagnostics. It could be used in some other, scary Frankenstein technology also. So we need regulations, as somebody else was saying, to take care, to take enough precautions that this does not disrupt and create some monster out of us. But to give an example out here of the studies which are happening based on the CRISPR, there are 3 companies, if you're interested to work on CRISPR technologies, which is CRISPR Therapeutics, Intellia Therapeutics, and Editas Medicine as of now. And they have partnered with known pharmaceutical companies like Vertex, Regeneron, Novartis. And where are they trying to? Genetic disorders, obviously. You know, in beta thalassemia and sickle cell disease, there is a genetic mutation, and therefore the DNA is not proper. So if you're able to go in, excise that out, that particular DNA, and correct it, it is a wonderful thing. And as of today, we are still only in phase 1 or phase 2 of drug development. Same holds true. I'll be touching upon what is CAR therapy, immunotherapy, and bispecific monoclonal antibodies. But to say that different kinds of genetic diseases, the CRISPR technology is being utilized to kind of further the technology and see how best we can help the humans. In the last 5-7 years, in fact, in 2017 or 18, the Nobel Prize was given, which again I'll be mentioning. The idea came up that instance, when a bacteria comes in, when a virus comes in like a corona, it is not that the coronavirus is killing everybody. Only 1.5% or 1.3 less than that succumb to corona. The rest of them are able to successfully fight it, or sometimes over successfully fight it, if I have to say. So our T cells and B cells identify what is a foreign cell and are able to kill them. A cancer cell, while developing, observes that whenever it raises its head, the T cell or the B cell is able to come and destroy it. So it becomes smarter, because evolutionarily, if you look at it, or clonal evolution wise, if you look at it, cancer cell is a far more evolved cell of our own. This cell understands that, look, these T cells and B cells are not killing our thyroid cells or liver cells or kidney cells or lung cells. What is that so special about them? So what they realize is that they are all wearing something like PDL1, instance, or CTLF4. What I mean by that is that they are wearing a red hat, for simplistic terms, let us say. So the normal cells, if they are wearing red hat, or T cell or B cell are not attacking them. So the cancer cell also goes ahead and wears a red hat, and thereby evading the identification by a T cell. Now, once our scientists discovered this, they said, can we create a molecule or an antibody which will go in and bind to this PDL1, thereby masking the red hat? So once the red hat is masked, then the tumor cell becomes vulnerable in nature. It is now exposed to the T cell, and T cell identifies it and takes it and destroys it. So that is the premise of immunotherapy. And as I mentioned previously, there are a lot of redundancies, multiple doors, multiple pathways. There's not just a single lock. So even though you kind of block this, the cancer cell has multiple other windows or doors open to escape the attack from the T cells. Or to use a very common analog, which I have to say. So let us say there is a Don. He's in a particular slum area. You get a warrant also to go and catch him. It is not easy. The moment the warrant is issued, there is a rogue policeman who is going to probably leak out the information to the Don that, look, they are coming to catch you. The rogue policeman, in terms of the human body, there are something called as, they're not rogue in nature, they're required to prevent autoimmune disorders like regulatory T cells or myeloid-derived suppressor cells. What are these? The job of these cells is to prevent indiscriminate firing by our T cells. So similarly, when you reach the Don, you know that he lives in a slum area. It is not easy to go there. Then roads get narrower, the lanes get narrower. So the blood supply will not be adequate for a tumor. The microenvironment for the tumor is not conducive enough. And therefore, sometimes the agents which you give will not go and reach there. Again, the reason for mentioning this is that this is how we understand regarding how certain patients do not respond. Even though there are spectacular drugs which we are creating, why is that the response rates vary only between 15%, 25%, 30%, 40%? Why is not the response rate 100%? And the reason for that is that these tumors have their own evasive microenvironment, macroenvironment, and other mechanisms by which they bribe the internal army inside us. I'll pause here. This is not just a talk on immunotherapy, but if there are any questions and somebody wants to drill down on this topic, I'll be more than happy to talk again. This is another fascinating instance. In terms of antibodies, we realize there could be monoclonal antibodies, then there are something called as bispecific antibodies, there are tetravalent antibodies, and there are quadrivalent antibodies. So in certain areas, what happens on a surface of a particular membrane, you require two agents to come together for a particular function to happen. In hemophilia, what happens is that factor 10, factor 9A do not club together because there's a deficiency of factor 8A. And the deficiency of factor 8 causes hemophilia. So Hemlibra is a molecule which has been created, which kind of binds with factor 9A and factor 10, thereby doing the function of what factor 8A does. And therefore, it prevents the bleeding per se. Bispecific antibodies have been developed even in hematolymphoid malignancies also. For CD19 binding, denosumab is one of the molecules which is there, which is in acute lymphoblastic leukemia. And similarly, we are now kind of trying to identify different therapeutic areas like multiple myeloma and other tumor types where we can use bispecific antibodies too. And before I move on to the next slide, I would like to just talk about, because I do not have a slide on CAR cell therapy, chimeric T cell therapy. The basic idea remains there that you are able to kind of take the antigen of the cancer cell outside the body and create more number of T cells and then reinfuse it back into the host cell. Thereby, now what has happened is that you have strengthened the army inside a cancer patient by developing the T cells from outside and putting that inside the, and that is what the CAR therapy is again. But the logistical challenges are very difficult. Only very recently, we had the first CAR therapy in India at Tata Memorial Center, which was done in collaboration with IIT Mumbai and Tata Memorial Center at Mumbai. Otherwise, these therapies are only available at few restricted areas for the obvious reason that it is not easy to kind of produce those T cells outside the body, which are having specificity and sensitivity, and re-inject it back to the tumor. And there are side effects. Since in the whole of this lecture, I'm not speaking about the side effects for different reasons. So we did see about a lot of pathways, a lot of interesting GIFs which kind of demonstrated, but I wanted to put a face, since by this time we would have probably got tired of the multiple pathways. And before I go to the individual people here, I would like to tell you that at least I know a person who was treated for vocal cord cancer in 1995, and it's almost like 30-40 years. And that was not with radiation, targeted radiation. So there are a lot of other technological advancements which have happened in cancer therapy apart from the drugs and the molecules. But that is not the focus of today's lecture. And then I have another 35, 36-year-old who got diagnosed with renal cell carcinoma around 2016. And unfortunately, he tried homeopathy for the first 6 months. So he got delayed, he got his surgery done. And then the metastasis happened. Metastasis is stage 4, where the tumor has kind of spread to the other areas of the body. What this resulted is that we had to start him on some molecule called as sunitinib. And since if you would have seen in the slides, it's a tyrosine kinase inhibitor, and sunitinib was a drug which was approved for renal cell carcinoma at the time. He took it for a year, but as I said, there are multiple pathways, multiple keys, and the cancer cells inside his body became smart, and therefore he developed resistance to sunitinib. We went ahead and tried pazopanib, but unfortunately he could not tolerate pazopanib, because all these pathways are also there in normal cells. And sometimes some people have diarrhea, some people have skin reactions, skin ulcerations, and they're not able to tolerate these molecules. Since he could not tolerate pazopanib, the immunotherapy which I was talking about, there was a nivolumab which was approved, and we used nivolumab, and for around 8 months he did well. But then he developed a mets in the brain, and then we had to treat those mets in the brain with something called as stereotactic radiosurgery, that is with radiation, and then put him on something called as axitinib. And with axitinib, again, he was doing well. But then there was another molecule called as pembrolizumab, which was approved. And then pembrolizumab with axitinib was used in him, and then he survived for another 1.5 years. Post 1.5 years, again, he developed resistance to these molecules. And today, with the knowledge that there was a molecule called as lenvatinib, he has been put on lenvatinib since the last 1.5 months. The reason for telling the story of this person is that this would have been a death sentence probably 20 years back in the first year itself. Today, with the advancements and the technologies which is available, what we are discussing, the kind of work which you are going to do in future, the kind of work which probably some of you are doing today, helps real people to live longer. And this guy is just 40-year-old as of today with 2 young children. So that kind of should be the motivation for us to kind of look into and develop new molecules which are going to come into the future also. The extreme left and the extreme right, what you see are the people who have been glorified in the last 1 year. and they are from the BioNTech, and the other one is from the University of Pennsylvania, and they were involved in the mRNA technology. In fact, the BioNTech were there to develop the cancer molecules, and if you visit their site, you will see that the mRNA technology is still being researched to kind of bring in effective therapeutics towards cancer too. While as the people who are in the middle are people who have been responsible for the CRISPR technology. The CRISPR, which I mentioned previously, is the upcoming one, and these people are the ones who received their Nobel Prize in 2020. And the ones who are there, Mr. Professor Tasuku Hanzo and James Allison, these are the two ones who were responsible for elucidating or making us understand how the immunotherapy escape mechanism or checkpoint inhibitors make an impact in cancer treatment. Shifting the gears, since we have been speaking about the different pathways, molecules, drug development, but all this is also facilitated by some other advancements in the technology which need to happen. How do we know at what stage the tumor is, how big the tumor is, whether the tumor has responded or not? You're not going to take a biopsy each and every one time. So one of the images which I've put across the screen is of a PET CT of a patient with clear lung. Previously, what I have highlighted in red, that used to be considered as tumor, whereas on the right-hand side, what you see is just the glow, what the tumor is all about. But this is not just to highlight this. Instead, it is to give you the kind of an understanding that the discovery of positron, which led to the identification of a PET technology per se, came from a mathematical equation which was written somewhere in 1920s by someone called as Paul Dirac. And it will be, if you are a fan of Beatles, you will find it fascinating that probably the success of Beatles kind of led to this discovery of CT scan. The company which produced the label of Beatles, EMI Records, were the initial pioneers of CT scan. So there are myriad ways in which the advancements and the help can lead to discoveries. And today, PET CT is an integral component of cancer imaging, even at the diagnostic level as well as the research level. Then similarly, what I have identified here is that in breast cancer, we know that hormone receptor positive, estrogen receptor positive, we treat them with anti-estrogen therapy. But at times they develop resistance, and what you see is that you are unable to kind of know which molecules or which pathways which we previously highlighted are still patent enough or are they resistant to. In the initial phase, you find that it is the estradiol receptor positive ones where you see a complete response, while as the ones where the estradiol negative one scan was there, you did not see a response at all. So these again help in drug development from the perspective that your successful launch of the molecule depends on having this kind of technologies at your disposal. Taking a pause again, all that we do today, we stand on the shoulder of the giants, and standing on the shoulder of the giants mandates that even as we step into Industry 4.0, we look back and see that from 1900, how the animal models to the first discovery of nitrogen mustard, which happened in the Second World War period, because the chemical warfare happened, and then we discovered that the red cell count goes down because of that. And that led to the first chemotherapeutic agents and the first successful cure of something called as choriocarcinoma using methotrexate. The Human Genome Project also was responsible kind of leading to the foundation for all that which we spoke in the last probably 35 minutes, I guess. So the highlight which is there on the slide is to say that gradually we kind of went ahead curing, whether it is testicular cancer, whether it is cure of ALL, whether the NCI investment in molecular biology, and there is a lead time of around 20 to 30 years for this to kind of bear fruit. All that we see today would not have been possible without the kind of investment in the basic research as well as the applied research in the last 30 to 40 years. And today we are able to kind of reap the benefits of molecular profiling, genomic sequencing, as well as the diagnostics, because this kind of investment has been made. To give a glimpse regarding the kind of changes which have happened, if you look at the first arrow, the discovery of the BCR-ABL as the reason for CML happened somewhere in 1960, while as the molecule only came in the era of 2001. That means it took us around 40 years. And then you will see that from Herceptin to its commercialization marketing, it took 13 years. For PARP inhibitors, around 15 years. For BRAF inhibitors, around 8 years. And for ALK inhibitor, which is crizotinib, it is around 3 years. So we have been reducing the time required for the discovery from the bench side to the market. But at the same time, this probably has been facilitated because of the Moore's law. But then there is something called as Irum's law, which says that it is getting costlier and more difficult to kind of research drugs. And you can probably Google it later and understand it better. But what I wanted to highlight is that with the advent of technology, you are identifying more number of locks, you are identifying more number of keys. But to bring them successfully to the market, as Professor P. Suresh also highlighted, only out of 25,000 to 50,000 molecules, one comes into the market. And why is that? You know very well, in sense, there are redundancies, there are resistances, there are side effects which are responsible. And there's not a slide which I wanted to highlight. Most of the molecules in the phase 1 fail because there are side effect concerns which are there. Whereas in the phase 2, where 75% of the molecules have to be dumped, it is because the efficacy which is there is not commensurate with what the industry kind of looks at it. We did hear about the AI ML automation by the previous speakers, and I am just pointing out here regarding the partnerships which the major pharmaceutical giants have made. Roche has a partnership with some organization called as Okin. Pfizer has with Extelpi. IBM Watson has been there for some time. Novartis has made partnerships with them. Bayer, Sanofi, Takeda. So these are all from the different periods of the drug development to the fact that today even the recruitment of patients, whether in phase 1, phase 2, phase 3, digitalization, digital health, monitoring at home, risk-based management, these are all the terminologies which are very commonly used and which you will come across. So it is basically to say that, can we reduce the cost of producing one molecule, of one successful molecule? Can we reduce the timelines of producing one successful molecule? And if COVID has taught us anything, it probably is that it is possible. It is just that we need the kind of the willpower which we had during the last year's vaccine development time. And then maybe with the help of the AI, ML, and the other automations which we saw previously, we should be able to kind of bring about successful drugs faster into the market using the CRISPR and other things. Again, I leave you with that image, and thank you for your attention. I purposely have kept around 10-15 minutes for any kind of questions which any of you may have, and I'll be happy to take your questions and answer them. Thank you.
Thank you so much, sir. It was indeed very, very enlightening. Thank you so much, sir. It was indeed very, very enlightening. That was really nice how you touched upon at cellular level, and I'm sure it was really insightful for the students, at a deeper level, and how you also touched upon AI, ML automation, and the kind of collaboration the organizations between pharma and tech organizations are having at the moment. And the question you rightly put right before you ended on, can we decrease the cost and the time that it takes to bring about new drug into the market? So there are lots of questions. One tick, sir, is a student asked, how far tissue-specific immunity will affect the efficacy of monoclonal antibodies? This is the first. So what is the view on it, sir?
Tissue-specific? Sorry, let me repeat the question for me.
How far will tissue-specific immunity affect the efficacy of monoclonal antibodies?
See, monoclonal antibodies are created against a particular target. For example, rituximab is against CD20. Okay, and we have for CD30 brentuximab. The whole intention of creating this is that we have a targeted therapy. So if I understand the question, the molecules go and bind to them. You may not find the same kind of response in all 100 people to whom you give them. The reason being that, as I said, cancer cells are also smarter in nature, and there would be other pathways through which they would escape this particular cell killing. But otherwise, with the targeted therapies, we have had fantastic response rate, and we continue to kind of improve upon them and understand how this affects. But I'm not sure whether I answered. The person can go ahead and ask a follow-up question, because I don't think I understood the question properly.
Okay, of course. And also, you did also touch upon that, how drugs act differently on different people, and that's all to do everything at the cellular level of each person at large. Another question, which is rather complicated for me, honestly, to understand, but I'm sure it is a smooth cake for you. So the question says, given the genomic data available with oncology drugs, given the genomic data available with oncology drugs, can they be repurposed for antisense and stealth targeting of viral targets?
Yes, it has been. In sense, the molecules which have being used to kind of use against cancer in the recent COVID thing, there have been certain areas where repurposing were done. But repurposing of drugs is not a one single day. One of the reasons why it is tempting to do repurposing is that the safety data of some molecules are already well established, and therefore they could be used. But you will have to understand that virus has come into focus because of corona. Otherwise, we have a joke. Let me say that joke for the rest of the my colleagues and the fellows to understand. When you have a cold, if you don't take treatment, it goes away in 7 days. And if you take treatment, it goes away in 1 week. That is the truth about viral infections per se, at a larger concept for the cold perspective. So Spanish flu, which happened in 2000 and since 1918, at that time it killed many. And today also influenza kills many. 3 lakh people still die out of it. This is a vulnerable population. So COVID will also get normalized in the next 2-3 years. We will develop our immunity. It will be fairly okay. The kind of viruses which are far more difficult are the hepatitis C virus, hepatitis B virus, and then the HIV virus kind of stuff. And we have developed quite significant good amount of medications, and Indian generic pharma has been at the forefront. In fact, has been lauded by the WHO also. So those kind of antiviral therapies are of a different nature. When we come from the malignancy perspective, I don't think the ability to kind of use it against the infectious agents is going to be that prevalent in nature. What would probably happen is that understanding of the RNA, DNA, and how we can probably insert the RNA to the DNA or change the DNA structure is going to improve further and further. And this probably could be used as a Trojan horse in future against some infections.
Thank you so much, sir. There's one more question. So the next question says, will monoclonal antibodies like pembrolizumab be effective in NSCLC or melanoma patients with liver metastasis, as liver itself has its own tolerogenic mechanisms?
They have been. In fact, if you look at it, these were first approved in metastatic setting in both non-small cell lung cancer as well as melanoma. In fact, melanoma is one of the first tumors where immunotherapeutic agents are kind of tried because it is more immunogenic in nature. All tumors are not equally immunogenic. Some have a higher tumor mutational burden, or some have high tumor infiltrating lymphocytes, and they are more immunogenic. And there, your pembro, nivo, as well as durvalumab act better. The lesions in the liver are not going to escape from the T cell, B cell attack just because there's a protective mechanism inside there. The fact that you are able to use the pembrolizumab and then it is able to take out the red hat, which I mentioned previously, will make the T cell, B cell go and attack the cells which are there in the liver. The challenge is that sometimes the side effects can be that our T cells will attack even the normal liver cells, causing hepatitis. So under those circumstances, we will have to be very careful and see how this particular side effect needs to be controlled. Otherwise, it could be a permanent hepatitis, resulting in catastrophe for the patient. Out of 100, around 25 to 30 respond to these agents, whereas the rest of them don't. And as I mentioned previously, because they have got escape mechanisms in place.
Right, sir. So there is a very, and I believe that would also be a very generic question for students nowadays, because we are told a lot of course, when it comes to tech and pharma integration, they can think about it at machine level, by which I mean, at development level. But when we talk about AI and when we talk about ML into pharma or targeted therapy, right? How exactly is it being used is a very generic question that students have, which one of the student has also asked over here, is how machine learning can impact a targeted therapy. How, they told a lot, but maybe they just want to understand a little more how of it.
No, I didn't go into the how. I'll be honest
Yeah,
with
exactly.
you.
They would
Some
like
of the slides
to,
also I just touched upon from the perspective.
Yes.
Because I would rather leave that to my technology guys.
Okay.
What one needs to understand is that machine learning, AI, even though the terms are very routinely used and more often used, and probably we'll hear it in the course of our conference today and tomorrow, you need to understand that it is a system created by the human beings at the end of the day. IBM Watson came up with a lot of hype, but unfortunately, it has not been able to kind of survive in terms of at least trying to define which chemotherapeutic regime has to be given to a particular patient. The promise was that it will read through multiple journals, multiple new publications much better than human beings, because the capacity of human beings to read and understand is far lower than that of a computer. But as of today, the regimens written by human beings score over that provided by the IBM. Having said that, it has had a success with the drug discovery and the genomic part of it. Our ability to understand the different genomic pathways and make sense of it is going to be limited. But coming back to the question, as a person who is, I am an oncologist, I have studied oncology, I understand cancer better. When I was partnering with IBM, what they required from me was the domain expertise. I am supposed to give them regarding what particular tumor, how the patient behaves, what is the pathology, how is the immunohistochemistry marker, which molecules succeed, which molecules do not succeed, which combinations we see. So a lot of data inputs have to get into the system first before AI or machine learning is able to give us any suggestion. There's a very old saying, garbage in, garbage out. What I mean by that is that unless and until there is a lot of data already fed into this system, the ability of the machine to learn and then give us answers is going to be limited in nature. And when we say that, we have to humbly accept that only 2 to 3% of the population participates in clinical studies or clinical trials, which is the data through which we speak, we talk about evidence. 95 to 97% of the population is real world evidence, where today the data, in sense any patient does not have the data stored on the cloud. It is not accessible. There are HIPAA laws, there are privacy laws. So I don't know exactly how a general lay public outside the trial, when he took a particular drug, how did he behave? That's where that pharmacovigilance comes in, pharmacogenomics comes in. And there are a lot of crosstalk which has to happen. And this data cannot be just unstructured in nature. So as a pharmacy student or as a medical student or as a healthcare worker, what I need to understand is that I need to develop a habit of creating and storing data in a proper fashion. All the patient data, all the molecule data, whatever data which I've got needs to be collected and stored in a better manner. We don't have that robustness in our country as of today for each and every molecule which we give. Once that data is there, then you feed it and also record the outcome of the person. Today, what Google Health and Apple Health probably are trying to do is that as people take these molecules, is their ECG changing? Are they having falls? Is their blood pressure falling? Are they having tachycardia? Is their heart rate going up? All are these things which needs to be kind of measured. So once you have the patient-related data, you have the molecule-related data, and they all combine together and they talk to each other, it is going to give us the inputs. So I don't recommend our students to go ahead and put, take courses in ML or AI, unless until they want to change the line. If they become domain experts, and if they can work with technology colleagues, that would be the best foot forward according to me.
Thank you so much. I was just writing that down. It was a beautiful point, and that will be my end part of the question today. But before that, I have a few more. So there's another question, Siddharth, who asks, what are the applications of Moore's and Irom's law? So that's a direct question, sir.
Moore's law comes from the technology. That is, every 18 months, the chip size becomes half of what it is, or the memory capacity doubles. In fact, what we have on our mobile today, the reason why we are able to kind of talk like this used to occupy large buildings when the rockets were first launched. So the computing ability has kind of doubled every 18 months for the last 30, 40 years. I don't know how long it can keep doubling, but there are plans to store this kind of data even on DNA. That is what Moore's law is all about. And that is what has created miniature computers inside in our hand from whatever mobiles we use. Coming to Irom's law is to only highlight that the cost of or the failure of creating a particular molecule is high, and we need to kind of borrow advancements in other areas to kind of prove Irom's law wrong. And I think the development of vaccine last year is a kind of defying the Irom's law. Irom's law is more to kind of say that the pharmaceutical industry, the pharmacists have to be aware of it so that we don't, get lost in the multitude of molecules, multitude of targets. We are more wiser in terms of how we identify them At least from the practical industry perspective, return on investment, the return on time invested, as well as the return on capital invested, are two things which need to be kept in mind to kind of bring about better results. That's what I would say.
Thank you, sir. Another question is, can we predict the patient response to checkpoint inhibitors?
Yes. So that is what is called about biomarkers. I did not go deep into the biomarkers. There are another group of companion diagnostics. So when you look at a person, you say is capable of doing X, Y, Z, or etc. So based on certain characters, we kind of understand that we can predict certain responses. So when you take a tumor, you biopsy them, and since you take a piece of it, then you study which receptors are expressed, which are not. So there is something called as combined positivity score. That is the PDL1 score on the tumor as well as the T cells. So you have more the expression. So can be 1%, it can be 10%, it can be 20%, it can be 50%. Greater the expression, the greater the response rate in some tumors. It is not all tumors. In NSCLC, the Kaplan-Meier curves separate in a different fashion at a particular percentage. In urothelial cancer, it is different. In melanoma, it is different. So it is not a universal biomarker. Tumor to tumor, it changes. Similarly, there's something called as tumor mutational burden. There's something called as neutrophil to lymphocyte ratio. So if you go to other tumors, there could be specific EGFR receptor positivity in terms of lung cancer. So multiple things are studied, and then you kind of come to a particular idea that this particular patient is going to respond to a particular drug. With respect to immunotherapy, we have a long way to go. And since we are not that happy, or I cannot say that confidently that a patient has come and there's a score, I would rather probably, in spite of the score being low, because these are used almost at third line, fourth line, and if the patient can afford, these are highly prohibitively expensive therapies. So what we do is that, let us say, give 2 cycles or 3 cycles, and if the patient responds, we continue to give them irrespective of whether the score is high or not. The example of a person whom I gave, the 35-year-old, 40-year-old who is now 40-year-old, he didn't have a high CPS score. If we would have relied only on his score, he would have been denied of that immunotherapy. But immunotherapy did benefit him irrespective of the score not coming high. Just to give the complexity of the biomarkers which we use for immunotherapy.
So, sir, of course, large part of our audience today are students, right? And very aptly, you touched upon, what's really confused, because of course, this is a place where they're confused. Should we now start educating ourselves in machine learning, in AI? Should we start taking courses towards that direction? How is it really, that it's going to benefit us? So you really touched upon a good thing that you said: become your domain expert. Right? Understand the technology, but become domain expert. And also a very simple thing that you touched upon, which we miss out, is being able to store and create and store data properly, that we do not really do. And that's, I think, the grassroots level from where anything related to tech starts. It's about data. It's about being able to feed in good data, right? That could give us the outcome that we want. So to the students, sir, who are listening to you, right, what do you suggest the med student, the student from the medical, paramedical fraternity, okay, who are really confused right now because of the tech revolution that's happening and everything is getting interlinked to each other? What do you suggest to them? What would be something that you feel could help them if they follow in terms of educating themselves, in terms of preparing themselves for the kind of advancements we are headed towards? How could, as students, they move forward, sir?
Thanks. So I think each, if you're only talking about even pharmaceutical industry, I think there are multiple sub-domains inside the pharmaceutical area. And whatever you're passionate about, and wherever you think you can spend hours together without realizing that you've spent hours together, dwell deep into it, become the kind of the domain experts required, whether it is pharmacokinetic, pharmacodynamics, whether it is drug discovery, whether it is drug production, commercialization aspect of it, or whether you want to get into the marketing aspect of it, irrespective of the kind. Don't think that one has got a greater, job prospects or something like that. If something is of greater interest to you, please put your all efforts into that, and you will probably get some opening sooner or later in that particular area. So becoming the expert in what you like, and then utilizing and connecting that across the different functional domains would be the crux of what you do in the next 10, 20 years probably. You need to be a jack of all, but at the same time, the mastery over one topic helps you to kind of increase the range across multiple things. In fact, if people are interested, they could read a book called as Range by one Mr. Epstein, which goes on to answer the specific question which you have asked.
Great, sir. Thank you so much. And I'm sure students, so if I'm sure you guys are listening, please do follow, do get the book, read it, right? Because it would help you a lot, because things are really getting interlinked, and it's important to understand the correct way forward, or detangled way forward rather. So before we end the session, your final thoughts that you have, that you'd like to tell us, like to share with us before we depart, sir.
Thanks a ton for this, organizing this and giving me an opportunity. What I regret is that I cannot see the faces of each and every student, which I would have loved to. And since that's always an energy booster for us, hearing the questions or interacting with them is something probably we will have to forego this year. Wish them, each and everyone, to stay safe, stay healthy, and believe in science. What I mean, in fact, the terminology believe in science is wrong, because science is full about data. Focus on the data, try to develop capabilities where you're able to read data in a proper manner, understand the data in a proper manner. Even in the educated people, I see vaccine hesitancy. People think that vaccines will harm them, cause them XYZ. Once you understand the technology behind it, once you understand how this mRNA or the killed virus, it goes inside and creates immunity, I don't think vaccine hesitancy should be a concern. It's the duty of each and every healthcare, pharmaceutical, life science student or worker to kind of ensure that the lay public is made to understand how the pharmaceutical industry works, what we strive for. And it is that we enter this to improve the lives and health of people. Not believe in some conspiracy theories that Bill Gates wants to insert something or China wants to do something. So the having an open mind requires that we are able to have an open mind towards all the information which comes in, critically evaluate it, develop the capabilities to critically evaluate it, and then take that information across to it. So here I would like to probably, and since my colleagues here would probably laugh at me, but there's someone called as Khan Sir from Patna. If you can watch his videos and take inspiration, it's amazing the way that person kind of is able to assimilate the data and bring it across in a manner which is easily understandable by the lay public. He should be an inspiration to each and every science student, according to Khan Sir from Patna. You can Google him, and I think you'll be able to enjoy his videos.
Sure,
That's
sir. Of course. Thank you so much, sir. Thank you so much. And it was indeed a very, very knowledgeful session, and a justice to the topic, technology and targets, reshaping cancer treatment. A lot of really technical questions, some really general questions. I'm sure it was really beneficial to people at all level, our audiences at all level. Thank you so much, sir, once again. And
My
we
pleasure.
hope you are with us next year. And it's always a pleasure to have you with us. So thank you so much, sir.
Thank you, Dr. Thank you. Thank you, everyone.
Thank you, sir.
Our next speaker for the day is Dr. Onkar Prakash Kulkarni, Associate Professor at Department of Pharmacy, Birla Institute of Technology and Science, Pilani, Hyderabad. He is presently working as Associate Professor and has received his PhD from University of Munich, Germany, and Bachelor of Pharmacy from Shivaji University, Solapur. Sir has more than 18 years of experience in the research field. He has worked as research internship at Zydus Research Center, Ahmedabad in 2003-04, as research associate at Department of Pharmacology, Ranbaxy Laboratories, Gurgaon, India during 2006, as doctoral thesis at Department of Clinical Biochemistry with the research group of Professor Andrews in 2006 to 2010. As postdoctoral research fellow at Department of Clinical Biochemistry with the research group of Professor Andrews in 2010 to 2013. As assistant professor, Department of Pharmacy, BITS Pilani, Hyderabad campus in 2013 to 2018. He has published more than 46 research publications having 3,363 citations with 36 H index of 36 and 46 I-10 indexes of 46. It is an honor to have you with us, sir. I leave the floor to you. Please enlighten the audience.
Thank you. Thank you for your nice words and introduction. Good evening, everyone. I have been working with the renal disease and the therapeutic targets for novel therapeutic targets for renal disease for most of my research career. I did my PhD in glomerular nephritis, which is a complication associated with systemic lupus. And after working with extensively with SLE as a nephritis model, I also had my exposure or work with ischemic renal injury as well as other renal fibrosis models. So currently at BITS Pilani Hyderabad campus, we are continuing, I'm continuing with my work with the renal field or renal inflammation field. And in today's talk, I will briefly talk about what are the different or what are the upcoming novel therapeutic targets for renal fibrosis. And as a lab, what we are trying to contribute to the ongoing research. So I'll share my presentation now. Okay, can you just acknowledge whether you can see my screen? So today's talk is about novel targets for renal disease, and in particular, I will talk about renal fibrosis, which is a chronic renal disease conditions. So when you define renal injury, one can categorize them as pre-renal injury, intrarenal injury, and post-renal injury. That means if, like, for example, there is reduction in the blood flow to the kidney, or the kidney is experiencing ischemic condition, that is what we call it as a pre-renal injury. So there is nothing wrong with the renal function or renal as an or kidney as an organ, but the blood flow to the kidney is reduced because of the vascular complications, and that comes under the category of pre-renal injury. Then you have intrarenal injuries where, for example, in diabetic nephropathy or crystal-induced nephropathy, where the high blood glucose levels, sustainedly upregulated high blood glucose levels, or deposition of uric acid crystals or calcium oxalate crystals in the kidney, that is causing damage to the renal tissue. That is why it is called intrarenal injury. And then you have obstructive renal injury. That means nothing is wrong with the kidney as an organ, but the flow of the urine is obstructed because of the calcification or other obstruction of the urinary flow. And that is what we call it as a post-renal injury. So what I'm trying to say here is there are multiple ways because of which the kidneys might get damaged. But no matter what is the cause of the damage or cause of the injury for the kidney, you have a certain pathways which are getting upregulated, and those upregulated pathways, either it could be an inflammatory pathway or a pro-fibrotic pathway as well as regenerative pathway. The balance between these pathways defines the overall outcome. For example, if you have a block of blood flow to the kidney for a fraction of minutes, let's say a couple of minutes, and if you can bring back the blood flow to the normal within a couple of minutes, nothing goes wrong in the renal function because the renal tubules can sustain that sort of or that degree of hypoxia. Even though there will be inflammation that can cause acute renal injury marker upregulation, but overall there is no loss of renal function. But if you have a persistent loss of blood flow for a longer period of time, and that could then ultimately lead to tissue remodeling, which is otherwise called as renal fibrosis. So when you talk about renal fibrosis or tissue remodeling, there are three different molecular mechanisms which are important. For example, the participation of the renal tubular epithelial cells, participation of the resident renal fibroblast, and the infiltrating macrophages and other immune cells. So what do you see in renal fibrosis or chronic renal diseases? You have epithelial injury, you have fibroblast proliferation, and you have macrophage infiltration, right? And these factors together, when they happen, for example, the macrophage infiltration will be responsible for upregulation of factors which are responsible for fibroblast proliferation. Once the fibroblast starts to proliferate, they will become active as well. So that is the transition of the fibroblast to myofibroblast. Once they become myofibroblast, they will start producing those extracellular matrix proteins, and that will cause the fibrosis. Right? Epithelial injury will release a lot of damage associated molecular patterns, and these damage associated molecular patterns then eventually participate in inflammation. At the same time, some of these injured epithelial cells will transform, or there is a term called EMT, epithelial mesenchymal transition, where the epithelial cells change their phenotype and become mesenchymal cells, become fibroblast like cells, and they start contributing to the fibrotic phase. So when you look at closely at a molecular pathways, you will realize that epithelial cell injury, fibroblast proliferation, and macrophage or immune cell or inflammatory cell activation, these are the key processes which are important for the progression of chronic kidney disease or renal fibrosis. And that is where, if you look at the clinical database, a lot of labs around the globe, they are trying to identify different factors which are regulating, for example, EMT, the factors regulating fibroblast activation and proliferation. At the same time, how can we reduce the input given by the macrophages? Like, for example, chemokine antagonists are being developed as a treatment for renal fibrosis, because by inhibiting the migration of these inflammatory cells from the vasculature to the injured renal tissue, you can reduce the extent of fibrosis. Right. So what I'm going to try here is I'll highlight important pathways which are participating in these different processes. For example, like I mentioned, when you talk about renal fibrosis is nothing but it is characterized as an excessive deposition of extracellular matrix, right? And that causes tissue remodeling, right? So you end up having a scar in the renal tissue, and that makes your renal tissue non-functional. And this particular process of renal fibrosis involves the participation of epithelial to mesenchymal transition, which is called EMT, as well as infiltration of monocytes and macrophages, along with T cell infiltrations, and then activation of myofibroblast. So when you look at these one by one, what is EMT? EMT, like I mentioned earlier, the tubular epithelial cells which are injured, they detach themselves from the tubular epithelial basement and they migrate to the interstitial space and transform themselves into matrix producing mesenchymal cells. Right, that's why it is called tubule epithelial to mesenchymal transition, and that is an important process where it contributes to around 10% of the myofibroblast population in the renal fibrotic tissue, and that involves disruption of tubular basement membrane, loss of epithelial adhesion, cytoskeletal reorganization, and upregulation of alpha SMA, which is a marker for mesenchymal tissue. And eventually, when these epithelial cells become mesenchymal cells, they start participating in the fibrosis by producing collagenous matrix. That's why EMT is an important process for renal fibrosis. Then you have macrophages and renal fibrosis, where activation and accumulation of inflammatory macrophages is one of the characteristics of fibrotic kidney. Right? And how do these macrophages participate? Once they start accumulating in the renal tissue, they produce different array of different factors, and these factors then eventually responsible for participating in epithelial injury. They also participate in production of TGF-beta, which is essential for initiating the process like EMT, as well as fibroblast activation. So macrophage do play an important role in the progression of renal fibrosis. And if you look at the clinical, what should I say, the attempt which are being done so far to reduce the progression of renal fibrosis, most of the companies are were heavily targeting on an important pathway which is central to the progression of renal fibrosis, which is nothing but TGF-beta pathway. So TGF-beta is known as a key pro-fibrotic mediator of fibrotic diseases. It can promote proliferation of messenger cells and also the matrix production. It also participates in excessive suppression of extracellular or what should I say? If when you talk about extracellular matrix, there are factors which promote ECM synthesis, and then there are factors which are called matrix metalloproteases, which are known to break down the ECM. That is how the balance is maintained. But what TGF beta pathway does, it suppresses the matrix metalloproteases and allows more and more extracellular matrix to get deposited, and that is why the fibrotic conditions get even worse, right? Epithelial to mesenchymal transition. So TGF beta is an essential pathway which participates, and if you look at the pathway carefully, the active TGF beta, when it binds to its receptor, it leads to the activation of TGF beta receptor 2 and receptor 1. There is a dimerization of these two receptors, and that dimerized activated receptor then leads to the phosphorylation of SMAD2 and SMAD3, which are nothing but the adapter proteins which get activated because of the activation of the receptor. And once the SMAD2 and SMAD3 is getting phosphorylated, it forms a complex with another adapter protein which is called SMAD4, and that SMAD2-3-4 complex then translocates to the nucleus. And once it translocates to the nucleus, it interacts with the different regions within the DNA structure and control the TGF beta responsive target gene expression, and that is how the TGF beta promotes the renal fibrosis. But when you look at the TGF beta pathway, it is also supported or amplified. There is a lot of crosstalk between the angiotensin 2 and its receptor and TGF beta pathway. There is also a crosstalk between advanced glycated products and its receptor and activation of inflammation along with or to that with TGF beta pathway. The EGF receptor pathway also interacts with or potentiates the TGF beta pathway activation, right? So there is a lot of interaction between or cross talking between the TGF beta pathway and other pathways which are important for renal fibrosis. What I'm trying to say here is TGF beta is a central to the renal fibrosis. That is the reason a lot of companies all over the globe, they tried their best to come up with the solution where they can interfere or intervene with the TGF beta pathway in order to reduce the progression of renal fibrosis. And some of these therapies I will talk about in next few slides. When you talk about the crosstalk between the other pathways and TGF beta pathway, there are some endogenous inhibitory pathways which inhibit TGF beta. For example, BMP7 is a protein. When it interacts with its receptor, it inhibits the TGF beta pathway. So when you look at the renal fibrosis, what you see, you have upregulation of TGF beta and you have downregulation of BMP7, right? And once that was identified, a lot of companies went on to work with identifying ALK3 receptor agonist, which is nothing but BMP7 mimetic, or a lot of companies also tried to inject recombinant BMP7 in order to reverse the renal fibrosis. So that particular data also I'll talk about in next few slides. So these are some of the therapies which are targeting TGF beta. For example, first two drugs are the TGF beta monoclonal antibodies. These are the antibodies which inhibit TGF beta. But if you look at the first antibody, LY2382770, it is against TGF beta 1 isoform. On the other hand, fresolimumab is an antibody which can neutralize all the isoforms of TGF beta 1, 2 as well as 3, right? Apart from these monoclonal antibodies therapies targeting TGF beta, a drug called pirfenidone, which is known to inhibit TGF beta expression and translation, and because of that it has been tested and successfully found to be effective in many of the chronic renal disease conditions like diabetic nephropathy as well as septic AKI or focal segmental glomerulosclerosis, right? But when you talk about the antibodies, which are the major attempts made by the pharmaceutical companies, when you talk about antibodies, there are some concerns when you are talking about anti-TGF beta antibodies, because TGF beta by nature is an anti-inflammatory cytokine. So when you neutralize TGF-beta, you are promoting inflammation within the body, right? So when you look at the TGF-beta, due to the multiple pathophysiological functions of TGF-beta, systemic administration of anti-TGF-beta antibodies may have significant side effects, given that the mice knockout of TGF-beta1 develops chronic inflammation. Like I said, TGF-beta is an anti-inflammatory cytokine, right? So if you have an antibody which completely neutralizes TGF-beta1, it promotes inflammation, right? At the same time, when you're talking about TGF-beta2 isoform, if you take into consideration the mice which are deficient in TGF-beta2, they die soon after their birth. So TGF-beta, even though it is an integral part of renal fibrosis, it is also participating in normal physiological processes. So depleting TGF-beta or injecting a long-term treatment with TGF-beta neutralizing antibodies might create side effects, right? So when we realized that, people started to look into alternative strategies. So one of the first molecule which against which the anti-renal fibrotic therapies were developed is called CTGF, right? CTGF is a cytokine which is thought to be a prominent pro-fibrotic downstream modulator of TGF-beta. And companies developed, and what we know about CTGF is overtly expressed in fibrotic conditions. At the same time, when you block CTGF by using antisense and oligonucleotides or antibodies against the CTGF receptor or factor, you end up having protection or amelioration of the progression of fibrosis. One such treatment targeting CTGF, which was developed by a company called Fibrogen, and the product is FG3019. And this particular antibody against the CTGF has been granted a US orphan drug designation for the treatment of idiopathic pulmonary fibrosis. Right. So targeting CTGF is another novel therapy which is under development for renal fibrosis as well. So how can we inhibit CTGF? One can develop antisense oligonucleotides. One can develop siRNA for CTGF specific siRNA, or an antibody against the CTGF, which is already been approved for as an orphan drug for idiopathic pulmonary fibrosis. Apart from CTGF, like I said, BMP7 is a negative regulator of TGF-beta. When I say negative regulator, what it means, BMP7 controls the TGF-beta pathway. And what happens in fibrotic condition, particularly in renal fibrotic conditions, BMP7 expression goes down. On the other hand, TGF-beta expression goes up. Right. So in order to bring back the BMP7 expression, what people have tried to develop different agonists like recombinant human BMP7. People have also developed the BMP agonist like BMP receptor agonist, which is THR123 or THR184. Right. They have also developed different regulators of BMP7. By injecting those proteins, you can upregulate the BMP7 levels. So altogether, many therapies were developed against or around the BMP7 upregulation, and some of these therapies have shown excellent outcome in the preclinical database or in preclinical trials. For example, this is the paper published in 2014, published by one of the leading researcher in renal fibrosis, Dr. Kaluri's lab. And what they have done, they have tested their indigenous THR123, which is a BMP7 receptor agonist. And what they have seen is when you use the agonist, it reduces the fibrosis in 5 different models of renal fibrosis. Like I mentioned in my first slide, there are different reasons because of which the fibrosis in the kidney develops. So what this lab or what this paper has done is they have induced the renal fibrosis in different models, and they have treated these animals with a BMP7 agonist, and they identified that this BMP7 agonist has a wonderful effect and it reduces the renal fibrosis in these particular mice. When you talk about BMP7, what BMP7 does, it binds to its own receptor and it also activates the SMAD pathway. But instead of SMAD 2 and 3, it activates SMAD 158. And what that SMAD 158 does, it causes inhibition of TGF beta mediated fibrosis. You're getting the point, right? So BMP7 activates a different set of SMAD proteins, and that utilizes SMAD4, forms a complex with the SMAD4, translocate to the nucleus, and causes anti-TGF beta effect. That is the reason BMP7 is called a natural antagonist of TGF beta pathway. So once we realized that interaction between BMP7 and TGF beta, a lot of companies went on to develop agonists for the BMP7 receptor, as well as some of the companies also tried injecting recombinant BMP7 and see whether the injection or the treatment with the recombinant protein is able to ameliorate or attenuate the progression of renal fibrosis, and that was successful. Apart from CTGF and BMP7, you have inflammation, which is an essential factor associated with renal fibrosis. Like I mentioned, inhibition of chemotaxis or chemokine network is known to have a significant impact on progression of renal fibrosis, like CCL2 or CCR4 antagonists or CCR1 inhibitors are effective in reducing renal fibrosis. Then you have phosphodiesterase inhibitors, because cyclic AMP upregulation in inflammatory cell is anti-inflammatory in nature. So in order to upregulate cyclic AMP level within the cytoplasm of inflammatory cell, what you need to do, you need to inhibit the phosphodiesterase enzyme, which are necessary for the breakdown of cyclic AMP. So these PD inhibitors are also known to have a significant impact or inhibition of progression of renal fibrosis. Then you have NOX1 and 4 inhibitor, which is known to participate in oxidative stress. At the same time, endothelin 1 antagonist and TNF alpha antagonist have also shown promising outcomes when it comes to inhibition of renal fibrosis. Now, there are some targets which are only tested or which are yet to be tested in clinical setup, but they are having an excellent data as far as the preclinical studies are concerned. For example, discordant domain receptor 1, DDR1 receptor, which is a receptor for collagen. So the antagonist for the DDR1 receptor is known to have a significant attenuation of renal fibrosis in animal models, right? Animals which are deficient of DDR1 expression are protected against the renal fibrosis. Similar to that, you have periostin, which is another molecule which have significant impact on renal fibrosis. Actually, periostin expression is in adult mice is reduced. It only gets upregulated during the inflammatory fibrotic conditions, right? And when you inhibit periostatin by using antisense oligonucleotides, or you develop the mice which are deficient in periostin expression, they are protected against the development of renal fibrosis. Similar to that, you have another target like connexin 43, which is a gap junction protein. Antagonism of connexin 43 is also known to have significant impact on renal fibrosis. Cannabinoid receptor 1, which is another important identification of pathway, because cannabinoid receptor 1 mainly participates in metabolic pathways. So cannabinoid receptor antagonists were initially being developed as a treatment for obesity, but what people have realized that the cannabinoid receptor 1 has a lot of side effects when it comes to its impact on central nervous system. But there are some antagonists of CR1 which are not capable of crossing the blood-brain barrier. When these molecules were tested in renal fibrotic condition, they have realized that cannabinoid receptor antagonism also attenuates the development of diabetic nephropathy and development of albuminuria. So these are some of the novel targets which have successfully been tested in preclinical setup, but their data about the human trial is still awaited. So when you summarize the progression of fibrosis, you have the participation of the epithelial cells, where injured epithelial cells release pro-inflammatory cytokines. At the same time, these injured epithelial cells also differentiate themselves from epithelial to mesenchymal phenotype and increase the myofibroblast numbers, right? Then you have macrophages which extensively migrated to injured kidney, produce different cytokines which are responsible for endothelial as well as epithelial injury. Some of these macrophages also polarize to form M2 macrophages, which are the major source of TGF beta in fibrotic kidney. And once TGF beta is formed, it promotes EMT. It activates resident fibroblast to become myofibroblast. And what is the difference between resident fibroblast and myofibroblast? The capacity to produce extracellular matrix. Once these resident fibroblasts get activated to form myofibroblast, they increase their capacity to form extracellular matrix, because deposition of extracellular matrix is the key event in the progression of any fibrotic condition, right? So this slide I want to explain, because in our lab we are trying to identify factors which can control macrophage activation, which can control epithelial mesenchymal transition, at the same time which can control the activation of fibroblast to myofibroblast, right? So when we did our research, when we did our literature review, we realized that there is one factor which has a significant impact on macrophage activation. It also known to participate in epithelial mesenchymal transition. At the same time, there are evidences where it also is expressed in activated fibroblast. And that factor is histone deacetylase 5. So from this slide onwards, I will talk about our own research where we are trying to understand the possible role of histone deacetylase 5 in the progression of renal fibrosis, right? So what do we know about in SDAC5? SDAC5 is expressed in renal tubules. So it is expressed by renal epithelial cells. SDAC5 acts as a modulator of regenerative response after the ischemic injury. So we know as much, or we know that after ischemic injury, the renal tubular epithelial cells downregulate HDAC5, right? And once the HDAC5 expression is downregulated in these renal epithelial cells, they become more pro-regenerative, right? What we know about the interaction of HDAC5 with BMP7, when you deplete HDAC5, you see upregulation of BMP7 in lung epithelial cells. But nothing has been known as of now about renal epithelial cells. Similarly, knockdown of HDAC5 is known to inhibit epithelial mesenchymal transition, which is induced by doxorubicin, which is an anti-cancer drug, right? So there is some evidence, or there is some glimpse of evidence about HDAC5 and its possible role in EMT, HDAC5 and its possible regulation of BMP7, as well as HDAC5 and its possible participation in macrophage activation. So considering those literature, what should I say, the points, we thought, why not go ahead and look at the expression of HDAC5 in renal fibrotic mice and see whether HDAC5 has some functional role to play in those conditions. So what we did, we tested HDAC5 expression in multiple different setups, and that is what I'm going to show you today as our own data, which is yet to be published. So what do we know? HDAC5 is upregulated in stimulated macrophages with crystals, because oxalate crystals induced renal injury is one of the common injury what you come across in India. Because of the hard water what we drink, there is a lot of calcium deposition in the kidneys, and because of that there is a lot of inflammation because of the active deposition of these crystals. So what do these crystals do when they deposit in the kidney? They activate the renal macrophages, and these macrophages become activated and they release a lot of inflammatory cytokines, and that is how you damage the kidney. You're getting the point. So what we did in our cell culture setup, we stimulated the cells or macrophages with media only, LPS only, oxalate crystals, and LPS plus oxalate crystals. So in this particular slide, if you look carefully, when you stimulate the macrophages with LPS and crystals together, you have a significant upregulation of pro-inflammatory cytokine, interleukin-1 beta, which is one of the most important cytokine for acute renal injury as well as chronic kidney diseases. The similar trend what we saw in TNF alpha, which is another important pro-inflammatory cytokine, which is upregulated in macrophages stimulated with LPS and crystal. Right now, in these cells which are stimulated, when we looked at the HDAC5 expression, it was significantly upregulated. Right, and an anti-inflammatory transcription factor which is regulated by HDAC5, KLF2, is downregulated in the same cells. So what you see HDAC5 is upregulated and KLF2, which is an anti-inflammatory transcription factor, which is downregulated. So here we could see that in macrophages stimulated with crystals, you do see upregulation of HDAC5. Now, this is what we did here was in vitro, where we stimulated the cells in the cell culture. We wanted to see whether the similar thing happens in mice which are having acute oxalate nephropathy. So in acute oxalate nephropathy, what we did, we induced the mice with oxalate nephropathy. And how do we confirm that the mice has oxalate nephropathy? Because the blood urea nitrogen, which is a marker for renal injury, is significantly upregulated in mice which are treated with calcium oxalate crystals. Right. So that confirms that these mice have oxalate nephropathy. And apart from BUN, you see pro-allone beta, mature allone beta upregulation is clearly seen. Pro-caspase 1 upregulation is clearly seen. And the similar tissue, when we looked at the HDAC5 expression, it was significantly upregulated. And KLF2 expression was significantly downregulated. So we could see the similar effect in vitro as well as in vivo, where in acute calcium oxalate nephropathy model, the HDAC5 expression was upregulated. So now we know that it is upregulated. These are the Western blot data about the expression of. So you can see here HDAC5 is clearly upregulated in mice treated with calcium oxalate. KLF2 expression is significantly downregulated. Caspase expression is significantly upregulated, both pro-caspase 1 and caspase 1. Right. So that is a clear indication that HDAC5 do participate or does get upregulated in acute oxalate nephropathy model. So now once we establish that, we wanted to see whether it has some functional relevance. So what we did, we developed a nano formulation of liposomes which are having HDAC5 specific siRNA encapsulated. So we made a delivery system of siRNA, HDAC5 siRNA with liposomal delivery system, and we injected those HDAC5 siRNA or exposed those HDAC5 siRNA liposomes in cell culture as well as in animal model. And what we see in cell culture, when you deplete HDAC5 using HDAC5 siRNA, the KLF2 expression goes up. Right. At the same time, the interleukin-1 beta and TNF alpha expression goes down. Right. So HDAC5 depletion inhibits macrophage activation in cell culture. Similarly, when we injected these liposomal HDAC5 siRNA in mice, what we saw, the blood urea nitrogen levels were significantly downregulated. Interleukin-1 beta levels in plasma were significantly downregulated. TNF alpha levels were significantly downregulated. And the kidney injury markers, KIM1 and NGAL, were also significantly reduced in mice treated with HDAC5 siRNA. Right. So that confirms our finding that HDAC5 do participate in renal injury associated with oxalate nephropathy. So this is another the similar data what you can see here, where we have done. So this is, you can see in mice which are having oxalate nephropathy injury, or the control mice, when you induce the calcium oxalate, the HDAC5 expression goes up. In HDAC5 siRNA, the HDAC5 expression goes down. Right. KLF2 expression, for example, is higher in normal mice it gets reduced in diseased mice and it is brought back to normal in siRNA treatment mice, right? So we did see some significant protection against the development of acute renal injury in mice treated with oxalate nephropathy. So this is the histology data which shows that the crystals are deposited in the kidney. These are the reasons why the kidneys get damaged. But when you are treating the mice with HDAC5 siRNA, the infiltration of neutrophils is significantly downregulated. Because any acute injury, you have neutrophil infiltration, which is a characteristic of the first phase of inflammation. And that is significantly downregulated in mice treated with or depleted HDAC5. So once we establish that HDAC5 have some significant role to play in macrophage activation associated with renal injuries, we also wanted to see what HDAC5 does in fibroblast. So what we did here, we established the model where the TGF-beta proliferates the fibroblast. So this particular graph talks about, as you increase the TGF-beta concentration, you have significant upregulation of fibroblast proliferation, right? But when you are inhibiting HDAC5 using HDAC5 siRNA, what you can see, there is a significant reduction of the fibroblast proliferation. So in this data, which is first of its kind, where we could see that HDAC5 is important for TGF-beta mediated proliferation of fibroblast, renal fibroblast, right? Now, once we know this, we wanted to see whether we could see also the activation of fibroblast inhibited. For activation of fibroblast, we used the marker, which is smooth muscle actin, and that smooth muscle actin was significantly upregulated in cells treated with TGF-beta. And you can see when you inhibit HDAC5, you can see a significant decrease in the alpha SMA expression. The similar thing what we saw in collagenase, where you have a significant reduction of the expression of the fibrotic markers when you are talking about HDAC5 depleted cells. So what we could see in this in vitro assays is HDAC5 is important for proliferation and activation of renal fibroblast. So now we wanted to see in a chronic fibrotic model of oxalate nephropathy, where the TGF-beta pathway is an important pathway. And what you can see here in chronic oxalate nephropathy model, after day 10 of oxalate nephropathy induction, you can see a significant upregulation of fibrotic markers like alpha SMA and collagen. At the same time, you see a significant upregulation of HDAC5 and downregulation of KLF2. So again, we could see that HDAC5 is upregulated in mice with renal fibrosis induced by oxalate nephropathy. Another way to confirm is, like, for example, now we wanted to see whether by inhibiting HDAC5, can we reverse the progression of renal fibrosis? Can we reverse the fibroblast activation? So what we did, we used PBA, which is a known HDAC5 4 and 5 non-selective inhibitor. And when we treat the mice with PBA, the blood urea nitrogen on day 10 of renal fibrosis condition was significantly downregulated. The creatinine levels were back to normal. HDAC5 expression was reduced in mice treated with PBA. KLF2 expression was upregulated. Collagen expression was downregulated. TGF-beta pathway was suppressed. BMP7 expression was upregulated. NLRP3 expression, alpha SMA expression, and FSP1 expression was significantly reduced in mice treated with PBA, right? So when we looked at the picrocytus staining, which is a marker for collagen, we could see there is a significant reduction of collagen expression in mice treated with PBA. The renal histology was improved. The tubular damage index was reduced, while there was no difference in the deposition of crystals. right? This is another Western blot analysis of when we see the BMP7, which is upregulated in treated mice, the collagen expression was downregulated, the TGF beta expression was downregulated, alpha SMA expression was downregulated, right? So this is again confirms that the HDAC5 inhibition does reduce the progression of renal fibrosis. So in the last attempt, what we did was we also wanted to see whether HDAC5 is participating in epithelial to mesenchymal transition. So what you can see here is the renal epithelial cells are stimulated with TGF beta. And what you can see, the HDAC5 expression is upregulated when the cells are stimulated with TGF beta. And when you are using HDAC5 siRNA, the expression of HDAC5 is significantly downregulated. And when we looked at the EMT markers, which is E-cadherin is a marker for epithelial cells, which is downregulated in the cells stimulated with TGF beta. But when you are depleting HDAC5, the expression of E-cadherin comes back to normal. So it is maintaining E-cadherin expression, right? Another marker for another important factor is BMP7. BMP7 in normal epithelial cell is upregulated, but when you stimulate the epithelial cells with TGF beta, the BMP7 expression goes down. But when you deplete HDAC5 and stimulate the cells with TGF beta, you do see upregulation of BMP7. So this indicates that BMP7 expression in epithelial cell is regulated by HDAC5, and HDAC5 inhibits the TGF beta mediated epithelial to mesenchymal transition. To summarize my talk, I would say that in our lab, we could see that HDAC5 regulates the macrophage activation. HDAC5 regulates fibroblast proliferation and activation, both in vitro and in vivo. And HDAC5 regulates epithelial mesenchymal transition, which is mediated by BMP7 expression in vitro. The in vivo studies are going on. We are about to complete those studies. And once that is complete, we'll also see the expression of BMP7 in vivo conditions of renal fibrosis. To conclude, we strongly believe that HDAC5 targeting could be a promising therapeutic option for renal fibrosis in the future. I would like to acknowledge DPT IYBA for funding the research, BITS Pilani Hyderabad campus for support and infrastructure, Pravesh Sharma, he is my research scholar who has developed all these models and tested HDAC5 expression and did all the studies what I just presented to you now, and my other laboratory members at BITS Pilani Hyderabad campus. Thank you.
Thank you so much, sir. It was indeed very enlightening and a complete justice to your topic, novel therapeutic targets for renal disease. And we could see your passion into the research that went into it, the kind of detailed explanation that we got. So it was really, really great, sir. Very, very knowledgeable for us. And we do have a few questions lined up, sir. So to start with, I think this is more of out of experience they would like to know, I guess. So the first question is, how effective are the novel therapies in treatment of renal genetic disorders?
Genetic disorders, I mean, like I said, most of the therapies are basically driven at TGF beta pathway. And it only helps you to control the remodeling what is happening along with the progression of the fibrosis. But for genetically predisposed conditions, like for example, take an example of SLE, what I have worked with, which also has a lupus nephritis, which is a significant renal fibrosis condition. But we cannot use TGF beta targeting therapies because that will increase the inflammation, which is even more problematic for a condition like SLE, right? So, I mean, you might be able to inhibit the progression, and that is the data also indicates, but one has to be careful because if the systemic or the genetic condition is gets or is influenced by the inflammation, which is influenced by the therapies, that would be a problem. But again, CTGF therapies are targeted for pulmonary fibrosis with the genetic predisposition, and they are shown to be effective. Similarly, BMP7 was tested in Alport syndrome, which is another genetic condition which is responsible for renal fibrosis, and it was found to be effective, but that too in preclinical stages. Similarly, you have therapies which are inhibiting tyrosine kinase or DDR1, for example. They were shown to be effective in Alport. Apart from that, the point what I missed in my presentation is a lot of labs are also trying to use mesenchymal stem cell therapies to treat renal fibrosis, and those therapies are also shown to be effective in some of the renal fibrotic conditions which are also having a hereditary background, like Alport syndrome, for example. So I cannot say in a generalized statement that all therapies are effective, but some of these therapies do have some impact on reducing the progression of renal fibrosis in such conditions.
Thank you so much, sir, for the answer. The next question is, are auxiliary peptides such as SMAD anchor or for receptor activation druggable targets?
I mean, the main problem with the peptide therapy is their, what should I say, their stability in the in vivo system. But since now we are developing a lot of targeted delivery systems, I mean, if you can use some targeted delivery, which can help you to deliver the peptides to the renal environment, they might be effective.
So it's still something that would be under research or under study.
No, there are many labs there are labs which are working on peptide delivery system, but I mean, I don't know whether they are only focusing on renal targeted, but there are definitely some progress being made to improve the delivery of peptides to the target organs.
Okay. So the next question is, how do we assess the druggability of proteins in silico for diabetic nephropathy?
Come again?
How do we assess the druggability of proteins in silico for diabetic nephropathy?
Proteins, you mean?
Yes, proteins.
Okay. But then again, like for example, BMP7, it's a recombinant protein, and it is known to get downregulated in cell culture conditions where you stimulate epithelial cells with high glucose. So it depends upon what model you are working on, right? For example, if you are developing a model which is more about epithelial mesenchymal transition or fibroblast activation, then you can use high glucose as a stimulus or advanced glycated end products as stimulus, which are usually seen with in hyperglycemic conditions, and then you can stimulate the cells. But again, the druggability will be a problem, because when you talk about recombinant protein therapy, one has to be careful about whether it has any non-specific interaction with any other receptors. Right. It is very easy to test the drugs or the proteins for their therapeutic benefits. I should not use the word easy, but it's relatively simpler to test the protein as a therapy in vitro conditions. But when you talk about in vivo, one has to be very careful about this recombinant protein is stable and it is not having any non-specific interactions. Its distribution one has to be careful about when you inject the recombinant protein, where it goes and binds and acts.
Thank you so much, sir, for that answer. The next question is again related to proteins. Are patients treated with tissue plasminogen activators such as alteplates are at high risk of renal fibrosis?
Okay.
So they would like to know what is your take on
No, I'm not really aware of that situation. I'm sorry.
Okay, sir. So also now another, I think, again, they would like to, I guess, know your recommendations. What would be the most important nutrition, because we're talking about renal diseases, so important nutritional recommendation for patients with CKD?
CKD, I mean, you have to be very careful about your electrolyte balance, first of all. I mean, a lot of CKD patients are advised not to have high potassium diet, for example. You also need to reduce the intake of certain food items which are known to promote crystallization like uric acid crystals, or like for example, some of the patients, I come across, they were asked not, because calcification is another problem which can also increase the renal fibrosis or CKD problem. Apart from that, one can use, because there are a lot of TGF beta inhibitors which are of natural origins, for example, curcumin. There are many such plant extracts which are known to have some impact on TGF beta pathway. So one can increase their intake, but of course with the dietitian in consultation, so that you can have some additive anti-TGF beta, because there are many papers you will come across where a lot of natural extracts are also known to have anti-fibrotic effect. So one can increase their intake, and that could improve or help in CKD. I'm sorry, I'm not a dietitian expert.
No. So I'm sure the question was also because you have such extensive research in
once you, it's like, a very narrow focus I have these days.
Yeah.
I haven't looked into the details about the dietary supplements.
More than narrow, maybe we would call it a more dedicated focus too, because the session was very informative in that sense. And now, since we're talking about drug development 4.0, right? So the next question that by default comes up is, what is your view on how efficient do you think AI or machine learning are going to be to develop newer drug targets in the future, or in this whole area in the future? What kind of effect do you see them having when we come to novel therapeutic targets for renal diseases? Let's also be specific.
One thing I see with respect to renal fibrosis is delivering a particular drug to the kidney needs to be improved. Because, for example, our lab is working on SDAC5, but SDAC5 is also important for in the physiological context. So going forward, if I want to really see its translational potential, I need to have some delivery system, and I also need to have some specific SDAC5 inhibitors. So you can, use the artificial intelligence to develop some sort of modeling system where you can have more selective inhibitors being developed, and come up with more selective and more specific inhibitors which can be developed for renal targeting.
So you do see a scope over there on making it more specific, increasing
we can use.
Okay. So I also from your bio can figure out that you do work with students. You're a professor yourself as well. So I'm sure you do also interact with a lot of students at your level.
Yes, I do. Yes. Part of my job is teaching.
Yes. So how do you motivate them? So because, the trend has become, like you said, very nicely, you have a very determined focus, right? And I believe that's something you might have also noticed missing somehow, somewhere, because there's just too much, maybe there's too much information, there's too much to know about out there. So how do you motivate your students? What kind of queries do you get from students in pertaining to, let's say, even research, if they want to really dedicate themselves into one area? Do you find those challenges? How do you motivate? If there are students listening right now who feel like I have interest in research, but I don't know how to start, what to do. So what do you suggest to them, sir?
See, what we do at our college is I encourage students to interact with the faculties in the first semester itself. If I see anyone from the first year in the corridor, I do interact with them, because, what I realized as a pharmacy graduate, one has to identify their interest as early as possible. Do not keep it for your third year or fourth year to determine whether I want to get into academics or industry or marketing or whatever field you choose. I don't really have any hierarchies or ranking for the choices they make, but whatever choice you make, you have to make it as early as possible. That's my recommendation to many of my students. Let's say by second year, they decide, okay, I want to get into biological research or drug discovery or formulation research, then they have ample of time to spend with the faculty in that particular area within the college and start working on projects. Right? There are a lot of PhD scholars, there are a lot of master students doing some sort of research. So what they can use that next 2 years is to learn the hands-on experience and to be a part of, even though it's a small degree, but we get involved with that process. Because what does it help is, it reassures them that this is where they want to go. Because a lot of the time it happens that they like pharmacology, but once they start working with the animals, they start or they start losing the interest. So if this is the scenario, it's always better to have that hands-on experience as early as possible, so that you are more assured that, okay, this is the field I want to get in. Apart from this, what I suggest, like for example, many of my students are somehow interested in neuroscience. And there is no such course in pharmacy curriculum which is dedicated on neuroscience. But now we are living in internet era. I encourage my students to do a Coursera course in basic neuroscience, applications of neuroscience, for which they can also get credits. Right. So many of my students have done 5-6 courses on basic neuroscience from foreign universities through open source platform. And apart from that, they learn the basic molecular biology or molecular pharmacology techniques with me. So by the time they graduate, they have enough theoretical foundation to get into neuroscience, and I help them to learn few techniques which are also useful for neuroscience. So when with that CV they apply for any master's program abroad or within India, the chances of them getting selected is even more. Right. So the neuroscience, for example, another field which a lot of my students are interested is diabetes. A lot of students are interested in cancer biology. But if you look at the curriculum, what we teach as a pharmacy student to a graduate, we only teach them basics of cancer biology, only a superficial introduction. But if really someone wants to become a cancer biologist or a pharmacologist working in the cancer biology field, they need to do some additional courses apart from their curriculum using the open source platforms and increase their theoretical base, because that is what is appreciated by the selection committee whenever you apply for the master's program. I strongly believe that, because that is how they see how that student has gradually developed the liking for that particular subject. So many of my students going forward, I mean, they have got admissions to master's universities or master's courses in abroad, and they're doing excellent. And eventually most of them end up doing PhD in other universities. So that is what I feel. If you are, and the same thing can be done for those who are interested in marketing and the finance or health economics. So a lot of my students do extra credit course for finance department. So there are some electives they choose. So they understand the economics, they understand the finance basics, and that is how they are ready for the job when someone offers them in the finance domain or marketing domain. So the key is to identify where you want to go as early as possible. And that is only possible when you start talking to the faculties in your department of different areas.
Amazing insights. So what I like the best, of course, start early, but what I like the best is about developing gradual interest, because many times we find that interest develops in students out of influences also.
Yes.
They see someone or they read something and someone tells them this is happening right now, you should have more work done in this area, and they tend to go regardless of what level of interest they might have into it. So I really like the fact that you said that being able to develop that gradual interest, and of course that would be by starting at the earliest, like you said. So if I had to really summarize it for the students' benefit, start at the earliest, make a gradual progression. So do projects, do hands-on trainings, whatever you can, so that you understand your liking, the depth of your liking, and don't restrict yourself. Use available, right? Online courses, anything that helps them grow better, and that could really help them develop the determined focus ultimately that it would be needed to come out with quality research and kind of research that can make difference and impact. Thank you so much, sir. It was really, really great to have you, to listen to you. Before we let you go, some final words for our audiences. What would you like to say? Anything, any food for thought that you'd like to leave them with?
I mean, the only thing I say, because we are all from the same fraternity, I mean, pharmacy students, I always see whenever they are asked, are you here by choice or by chance? I want to get the answer by choice, because, many a times, this as a profession is being sidelined for some reasons, I don't know. But there are so many things to do. I mean, you can do a lot of things being a pharmacist, being a pharmacy graduate. So I always, I want to, have that psyche changed. I mean, when the students come, and I see these days in our students, at least half of them are saying we came here by choice, and that's encouraging. So I really want the seniors to set that trend so that they can talk to their juniors and motivate them to do good things in pharmacy profession.
Thank you so much, sir. Thank you so much for giving us your time, and thank you for the session. Thank you so much, sir.
Thank you.
Thank you, sir.
Next speaker for the day is Dr. G. Jagdish, Senior Scientist at US FDA, Rockville, Maryland, USA. Dr. Jagdish received his undergraduate degree in pharmacy from the Government College of Pharmacy, followed by a master's and a doctoral degree from the All India Institute of Medical Sciences, New Delhi, and Banaras Hindu University, Varanasi, respectively. He was a reader in pharmacology at the Department of Pharmaceutics, BHU. He was a Canadian Heart Foundation Fellow at the University of Saskatchewan, Canada, followed by a Staff Scientist NIH Research Fellow at Northeastern University College of Pharmacy, Boston. Besides doing research, he also taught undergraduate and graduate classes at the university. Dr. Jagdish began his affiliation with the U.S. Food and Drug Administration at Silver Spring, Maryland in 1990, where he is presently a senior expert pharmacologist. He was an adjunct scientist at the NIH. His expertise is in cardiovascular and autonomic receptor pharmacology, G-protein-coupled receptors, and signal transduction. Dr. Jagdish has authored or co-authored several original articles, review articles, book chapters, edited 4 books, and commissioned several special issues for scientific journals. He is a peer reviewer for several journals, grant proposals, and thesis examiner for many universities. In addition to the above activities, Dr. Jagdish serves on several scientific committees, editorial boards, and professional organizations. Today, Dr. Jagdish will be speaking on development of potential repurposable drugs for COVID-19 based on the life cycle of SARS-CoV-2.
Good afternoon. I hope you are able to, I'm audible to everyone. Am I audible to everyone?
Yes, sir.
Yes, okay, thank you so much. I appreciate it. Good afternoon, and I'm very happy to be here at this convention. And I thank so much for inviting me to this forum this morning for us actually in the US. So it is late afternoon for you people. And being the last speaker, I hope I won't lose the audience while speaking on this topic. And also I thank profusely Professor Dr. Suresh Nagpal, Professor Karvekar, Professor Prakash Mallia, and also Principal Dr. Rajendra, as well as all the coordinators who made this convention a great success. Really, it is very tremendous. It's going on for the whole day, and tomorrow also it is being conducted. And I encourage everybody to take active participation and learn this emerging drug trends. Coming to my topic, it is development of potentially repurposable, and I will be able to share my screen with, okay. Just give me a second to start this one. Okay. It is, as you see the title here, it is potentially repurposable drugs. And for this purpose, I have chosen the current ones. That is, I know that whole world is in pandemic situation, and it is very disastrous to see that so many deaths are happening around the world. And I thought possibly this would be the right choice to discuss on what are the existing drugs could be drawn towards this disease COVID-19. They're the main purpose, so that I'm speaking on the current trend, which you will be able to listen for the next about 45 minutes from me. And also, if you have got any questions, please chat it in the chat box, or you can always unmute yourself and ask me the questions. Thank you so much. And my lecture outline is, first I will speak as before I introduce this topic of this array of drugs, because you know, the last several centuries, many number of drugs have been approved, and we are repurposing for this particular disease, COVID-19, caused by SARS-CoV-2. So in that case, definitely we should be able to understand what is this disease, what is this virus, what is its life cycle, because you would like to fit these drugs in the life cycle of the SARS-CoV, that is, from the way it gets attached to the host cells, that is, the host means here in the case human beings, the host cells, it gets attached. From there it multiplies and causes havoc in the body, inundating all the organs, the vital organs, and finally it kills the person in case the titer value goes up, and the sufferings. And what are the different drugs? Because we know that there are no drugs except the vaccine is the only one ultimate choice, which have been recently approved in the past 6 months. It is throughout the world, it is being given, and there is no particular drug for treating this disease. So we are repurposing from the existing armamentarium. So that's why I would like to introduce the virus, the SARS-CoV-2, and the disease caused by the virus, that is COVID-19. And that's why I would like to go into the structure and how it replicates in the body and how it is being released. The first 3 parts discusses for about 15 minutes. I may spend time here on this virus. Then we will go into the potential drugs for drug therapy. What are the purported drugs which we can be able to fit in the life cycle and supposed to combat the disease. Of course, a lot of failures are there, no doubt, but we could be able to see that how we are able to proceed with this one and what are the things it does it. We are divided into 2 parts. One is the virus host cell factors. Second part is once it reaches the threshold, it causes in the body, it sets in motion of cytokine storm, how that can be alleviated, maybe decreased to some extent so that patient can survive. That is the part we're discussing about second part. So these are the 2 main part about the repurposable drugs one. Then I summarize my findings. Now, what is SARS-CoV? It is more than an infection. We know that worldwide there are about 100 million cases or more is coming up, and death is about more than 4 million. And yesterday, India said that crossed 0.4 million, that is 4 lakhs deaths, and stands third next to US for the first one, second the Brazil, and third comes the India. It is very sad situation that the death rate things are increasing. Of course, I don't think, I think everyone is worried about much more part, especially this 2020. It has globally, it has affected much more than that of, that is 2021. First 6 months, it is greater than that of the previous, the whole year itself. It is still rising, and economic devastation on a global scale is very much large. And every nation is trying to control it and giving the vaccines. And I hope that it will come to an end before the end of this year. The top 5 countries which I already said, the US, Brazil, India, Russia, and Mexico are the ones. And in population wise percentage, if you take it, India is not that very high. There is one little bit consolation saying that we are not that very high if we take into the cases. The virus, how does it do? You know that it enters the body and spreads the disease and causes all these things, and that made people to come up with a lot of hypotheses, and experiments have been done on animals, as well as now they are doing a lot of clinical trials. You know, there are a lot of papers being published in the past 1 year, and recently someone has published a paper in pharmacological research in doing the bibliometric study of how many papers Indian universities and institutes they overwhelm, especially the review articles people doing with using in silico procedures and computer aided drug design. They use it to track down all of these natural products. Also they use it, and it is tremendous work has been done by Indians also in this case, and somehow it is not being picked up in the South India, especially by Karnataka and Tamil Nadu and Kerala. These three states are a little bit lagging behind in publication. Very trickling number of papers have been come out from these three states. Otherwise, upward towards the north, northeast, and also northwest regions, a large number of kinds, chemokines, which I'll be discussing in the last part segment of my talk. So let us move to the next stage, showing this how the virus. You see that the red one, the pink one is a cell, and the number of virus particles attached to this one is a multiple one. Similarly, there are about trillions of cells. If all cells are being attacked, how the person can survive? So it's a question of titer value. How many thousands of particles these are they multiplying very, very fast rate also. This is very impressive electron microscopic people taken and published this one, and you could see that how much the cell has been inflicted by this virus. Now, this is the structure, overall structure of SARS-CoV-2. Now, any layman knows about this one nowadays because the spikes, even the policemen, when they're on the road, they're putting on that helmet with the spike one to show that the spike one is which you're going to target and attack you, to show that one. And this is you see that it consists of about 4 structural proteins. Fifth one is not there, I have not included, which is hemagglutinates. And the 4 structural proteins, important ones is the envelope protein here. And there is a membrane protein, the nucleic acid protein, which contains the viral RNA and the lipid membrane, and the important aspect which attaches to the host cell is the spike protein number 2. And this makes up that 4 structural important proteins. In addition, it also contains 16 non-structural proteins, which will come across later on in my talk when I speak about the life cycle. 1 to 16, how this non-structural proteins also being reproduced. All this together, they're all multiplied in the host cell and then assembled together and then released into the circulation, and so that it can inundate other cells. And this spike protein contains 2 parts. See, I enlarged it here, S1 and S2. The outer part is the S1 subunit, which at the tip, it contains the receptor binding domain, this part which attaches to the host cell, and S2 subunit is able to get into that, it maneuvers such a way that it can enter the host cell. So attachment to the receptor is by the S1, and this has to be cleaved in the body. That is the enzyme which helps in cleaving this one. And you may wonder in your mind that how about if we prevent this cleaving? Prevent this binding to the host cell, don't you think you'll be able to achieve a great thing? Yes, that's why we're talking about certain drugs which would be able to attack at the initial stage of infection itself. How before it proceeds, getting into the body and endocytosis and then multiplication, then releasing. As I said about attachment, this is that attaches to the human ACE2 receptor, and this is the spike protein. This is a sequence of this starting with the S1, and this the tip of this one binds the number of amino acids on the ACE2 so that it can make an entry into the host cell. Now, this is based upon our recent publication, which we published in Cell Signaling paper in the last year. and I'll summarize the whole life cycle in this couple of slides. Of course, I might go into a little bit description also how this is being done, and with the whole summarizing. The first part you can see, follow this for 1, 2, 3, 4. The first part is, this is the virus with the spike protein S1 and S2 domain. As I was telling, that S1 is the main thing which attaches through its receptor binding domain, that is to the sites of this ACE2, that is the protease domain of the ACE2. This is ACE2 which having anchoring points, and the top it is having a very good site. One is the protease domain to which attaches this receptor binding domain of the S1 spike protein of the virus. Other is the catalytic site, which is a beneficial. This catalytic site, which of course I will explain a little bit later on also, this catalytic site which helps in generating angiotensin 1,7, a beneficial peptide coming from angiotensin 2, which is harmful. Of course, it has to be present in the body to cause vasoconstriction. Otherwise, blood pressure will not be able to maintain. But this catalytic site is very important, where it is able to convert that angiotensin 1 as angiotensin 2 to form the final product, angiotensin 1,7, which is able to protect the lungs as well as the heart, a beneficial one. And whereas the other part of this S2 as a receptor for the S1 domain of the virus to bind and initiate the process. Once it binds, then it is able to able to get into the body. Before binding, there are two membrane bound particles here, furin and TMPR SS2. These enzymes are able to prime this spike protein so that it is able to attach very nicely to the protease domain of the ACE2 receptor. So once it gets attached here, then it's able to endocytose enter inside. So you should have a potential drugs to block this whole process. Number one, and number two is when the endocytosis, which I'll be coming when I fit the drugs into this pathway. So pay attention to this life cycle of the virus. The intact virus getting bound to the ACE2 receptor, internalizes, and in the lysosome it digests and it gets released, uncoated, releases the positive, that is a single stranded RNA. This is a RNA virus, not a DNA virus. So single stranded RNA which releases, and this acts as a messenger RNA using the host template ribosomes, and it is able to form two polypeptides, that is PP1A and PP1B, as well as the protease enzymes 3CL Pro and M Pro. Whereas this 3CL Pro and M Pro, their protease is able to cleave these polypeptides to form 1 to 16 non-structural proteins. This is what I said. One of them important one is RNA dependent RNA polymerase NSP12, which is able to synthesize, replicate to form a more number of virus, and it gets assembled in the Golgi apparatus, and finally through exocytosis they are burst out and releases and infects the cells. And in fact, the first one part is the lungs, because it enters through the nose and the mouth, and also through the eyes it enters, and especially when it inundates the lungs, gets saturated, also gets into the next part is the heart, and then it goes to the kidney, brain, everywhere it in. So the first, the target is lung. That's why it is known as ARDS, acute respiratory distress syndrome it causes. So I hope all of you are able to understand this life cycle, how this is able to get attached to the host cell, then endocytosis releases the single strand RNA, which is able to be, and that becomes a messenger RNA and uses a ribosome's host ribosome, and translates and replicates, and using this important enzyme, which is the RNA-dependent RNA polymerase. Again, we'll come back to the drugs, which can attack on the fourth stage also. Then is packaging and viral assembly and releases through exocytosis process. Now, coming to the main topic about the repurposable drugs. Now, we have to see that what all the drugs can fit into this life cycle, as well as after it causes the storm in the body. Now, what is repurposable? Which is a definition, is an adjective word to be used if it's capable of being turned into another purpose. That is purpose, repurpose plus able. So we are abling these existing ones. Say turn out. That is, you say the old wine in a new bottle. So we are putting the existing approved drugs for treating a new disease. The drug repurposing defined as developing a new uses for the drug beyond its original use or approved indication, approved for one disease, we are extrapolating for the second disease, third disease, but because we already know their properties of the compound, it's CMC, that is chemistry, manufacturing, control, quality particles is already known, preclinical studies have been done, its pharmacology is known, toxicology is known, ADME part is known, and it is surveillance data by the previous approved one, it's approved by several agencies, so we know all of them. So only thing is it can rapidly enter the clinical trials by we can go to phase 2 and phase 3 immediately, so we can avoid, skip the line, this is what they say, skip the line, traditional line, and move to the next line. So repurposed and rediscovered drugs are regularly prescribed off-label. Even many of the drugs which physicians use, they always know that they are useful for another one, they use it, they call it as an off-label use. That is off-label use always encouraged, but the only thing is they are not being indicated for particular disease, but by the knowledge and extensive knowledge, physician always uses it. So that's why now for the COVID-19, these drugs are being repurposed and being drawn to this one new territory. Now, as I said, that one, it can skip this death valley. There is number of compounds which can pass through the initial stages from the discovery to validation, phase 1 trial, phase 2 trial. It can skip and it can move to the later part of the phase 2B, or it can also get into the phase 3 clinical trial. And it can cut down the cost involved. That is usually $2 to $3 billion if you move from the discovery stage to the final approval stage. And this number of steps can be skipped, so you can cut down the cost as well as number of years. Within 5 years, within 2 to 3 years, with a less cost like $300 million, one can get into a new one. And at the beginning of the pandemic, February 2020, there are only 10 repurposed drugs for under clinical trials for evaluation, and currently we got about 78 repurposable drugs belonging to the various classes, a number of classes, are now targeted against this virus COVID. Now network analysis, bioinformatic analysis, people have done it, this one, artificial intelligence, and then there is CAD. All these people are using it in silico methods, the way of methodically and rapidly in addition to purposeful candidates, they use it. And of course, this is one of the good papers where they have used this mapping of these protein-protein interactions. You know that for any disease, there is a proteins involved. The protein may be, that is usually it goes into disarray. So if you're able to tag that protein and connect this one, interact dome between that two proteins, and we got a druggable ones, proteins. In that way, they're able to come up with 69 compounds which can interact. Out of his 29 compounds have been approved, and 12 of them are in clinical trials, and 28 of them are in the preclinical compounds. And as you see in this interact dome, I hope, of course, it's difficult to see all of them given in one figure. You can always refer to this publication, Nature, which has come out where the people mapped the proteins, the virus protein. Remember, there are about 10 non-structural proteins, 16 plus another 4, 20 proteins. They are mapped to the host proteins, different proteins, come up with a drug. And there are about 332 interactions taking place between 26 of the 29 proteins where they are able to map it here, viral proteins. And there are about 332 human proteins connected to different diseases. And those, they made a very good interact dome and come up with drugs, you see. Now they fitted the drugs, all of them, these drugs here, with the green color ones. They are now the drugs, hydroxychloroquine, apamycin, and basitinib. So all of these drugs have been fitted here with experimental activities as well as the investigative drugs. In fitting all those things, they could come up with one drug, that is dextromethorphan, which you might have seen that the ingredient of this drug in the cough syrup. This is not against the virus, not antivirus. It allows the virus to grow better in the cells. That means the pro-viral effect. That's why this Gordon in the paper says, possibly also in one of the reviews, people have commented saying that, so dextromethorphan is a ingredient in the cough syrup, especially in the US. In the robitussin cough syrup, we use this DM, comes as a dextromethorphan, a very potent cough suppressant. Of course, codeine also we use in India, and dextromethorphan also being sold, I think so, in the cough syrup. That should be avoided. In such as you'll be having cough and other things at the stage, don't give to a patient dextromethorphan containing cough syrup, because it is a pro-viral effect. As per the mapping is concerned here, they show that it is not antiviral, it is a pro-viral. So it should be avoided. This is what the author writes in the paper. Now, how we can classify the drugs? They have said about 79 drugs. There are a number of drugs. People have also a number of drugs are in the clinical trials. They can be classified broadly into two categories. One is the viral host cell targets. That is depending upon the cycle, which I already explained in the first beginning 3 slides. That is its origin and the life cycle. If you have followed all the 4 and 5 steps in the origin of the life cycle, there we can fit this number of drugs here. And also once it is being multiplied and inundates the target organs like lung and heart, then it sets in the motion the immune response. Then what we want to do is the immunomodulators or anti-inflammatory drugs. That is the corticosteroids are the one being spoken here because they're already been approved. So these are the host immune response, which I'll be discussing this one. And also, just for the sake of discussing here, I'll bring up the neutralizing monoclonal antibodies. They are not repurposable. They're all laboratory made ones, which they'll be giving to the patient at the stage of treating them. And now, of course, these vaccines also will be acting like producing the antibodies, which is not the part of my talk, but I'll be just mentioning this one as a passing remark. So number 1, number 2, I'll be discussing these two in the next couple of minutes. Number 1, taking up this one, viral host cell targets. That is based upon the origin, the virus, and its life cycle in the body. Targeting the spike protein viral attachment to cellular receptor, targeting the ACE2, recombinant ACE2, endocytosis, going to the endosome, lysosome, releases, it causes proteolysis and translation, replication in the body. Okay, now let me get into this one step by step. Now, targeting the spike, the virus, it targets, attaches to the ACE2 receptors. This is the receptor portion of the ACE2. This one is primed before attaching with the furin and TMPRSS2. It attaches, it breaks down this S1, S2 units such a way that makes it easy for it to attach to the ACE2 receptors, and this turns down and able to penetrate the host cell. In this process, we got the drug camostat. Now, camostat prevents this furin and TMPRSS2. We got nafamostat. Both of them are being approved in Japan. They block the TMPRSS2 activity so that they will not be primed, so preventing the attachment here. And these are the protease inhibitors, which are also being used in various for the diagnostic tests as focused on the spike protein. All of them, these vaccines are based upon the spike protein. Now, next one is we want to deploy decoy ones. Decoy soluble ACE2, so that before it gets attached here, we will have a recombinant, human recombinant soluble ACE2 antibodies, so that this can go attached to spike protein, so that prevent them in attaching this one, so that better we can make use of this catalytic site to generate angiotensin 1,7, which is much more beneficial, so that we can spare these sites not to be endocytosed. And also spike vaccines are also based upon this theory of using the soluble ACE2 which circulates in the body, as naturally it also circulates, but very little. But if you could give exogenously, which will be able to act as a decoy and preventing the virus in attaching to the ACE2 receptor, that is known as the full length ACE2 recombinant ones. And other drugs which can also be useful is this acting upon the ACE2 expression. Some of them are also this renin, angiotensin, aldosterone system inhibitors like ACE inhibitors, aldosterone antagonists like eplerenone, spironolactone, statins, PPAR gamma agonists, glucagon peptides, DPP4 inhibitors like gliptins, linagliptins, beta blockers. These are to some extent people are already taking this one drug. They should continue to take, because that would be useful for them to have an ACE2 expression. Some people are discouraged now in the beginning process, but now it has been refuted that these people who are already having a secondary disease, comorbidities, and like diabetic patients, hypertensive patients, heart failure patients, they should continue to take the drugs because they are also useful to some extent. So statins and the expression of the ACE2. How does it work? Now, angiotensin 2 acts as 81 receptors, which is able to stimulate ADAM17 and which removes ACE2. But if you use ARBs, angiotensin receptor blockers, 81 receptor blockers, which blocks this process, blocks activation of ADAM17 and provides ACE2, not because for the binding, but because of for generation. This is what I said. Here arrow mark, you see the green one. It stimulates catalytic site to generate the more of, this is what I explained here, angiotensinogen pathway, ACE and ACE2, blocking ACE inhibitors or prevents this process of vasoconstriction and generates more of ACE, angiotensin 1,7, and they're able to stimulate the vasodilation, cardiac protection, anti-inflammation, anti-proliferation, anti-thrombosis. These green ones balances against the red one coming from angiotensin 2, that is a rebalancing ratio. What we want is the left side one, that is the versus right side one. So right side one, ACE2, should go up, whereas ACE enzyme should go down in such a way that angiotensin 2 decreases, angiotensin 1,7 increases. That process, if you could able to achieve it, that means we will be able to get this pathway upper hand. And this is a very tedious process because SARS-CoV-2 binds here, and whereas the beneficial part binds on the catalytic site. So ACE2 is a dual one. It is one side a negative, harmful by allowing it SARS-CoV to bind. Another side is a beneficial, allowing catalyzing, not binding, catalyzing here. It is a binding here receptor, catalyzing the conversion of angiotensin 1 to 1,7, again angiotensin 2 to 1,7, which is able to interact with another set of G protein coupled receptors, MAS receptors, and MALT receptors to have a vasodilation, cardiac protection, anti-inflammation. Thus, we are able to treat ARDS. Whereas this causes ARDS, this one, because angiotensin 1, 2, stimulating 81 receptors, it causes vasoconstriction, cell proliferation, inflammation, fibrosis, and decreases ACE2 expression. Now, this is the one which I explained in the previous slide, RAS inhibitors, that is which are useful. So people who are taking the effect of RAS inhibitors in the pathogenesis of COVID-19 is overly complex and controversial, but however, the American Cardiology Society, Heart Association, all of them said people who are hypertensive patients who are taking these ACE inhibitors or should continue to take this one, not to discontinue, other may be harmful, may be there. So this is what I discussed overall about the first and second step. In the second step, when it fuses, we have got to inhibit the membrane fusion by using hydroxychloroquine or Bidol. You know, hydroxychloroquine was in the picture in the last year when they said worldwide people started using it, but however, somehow it gave a very inconsistent results, and it was not uniformly. Some people praised it, some people said no, it is not, it is harmful and should not be taken, even though it raises the pH here, makes it alkaline, and acidic pH is favored for the virus, and that of course SCQ is having a multiple effect, not only this endosomal pH increase, and also it's an antiviral effect, as you see in the next one. It multiple effects. That's why in the beginning they were to give the healthcare workers as well as for the other people, frontline workers like police people and the sanitization people, all those people. But somehow, I don't know, somehow it did not work that effectively, mainly because of it's also having a cardiac effect and increasing the acute interval prolongation that might have precipitated much more. You can see I summarized that its mechanism of action, antiviral properties of this chloroquine is also having immunomodulatory effects besides alkalizing the vascular content. And also it's a zinc ionophore. Some people come up with a formulation, chloroquine with a zinc adding together. So that, and also it inhibits the glycosylation of the proteins here. Glycosylation is essential so that it can evade antibody attack. So it sugar coats glycolysis. That is also prevented by hydroxychloroquine. If you read all the mechanisms, which people have written a lot of reviews, papers especially come from Indian institutes and colleges, they put a lot of reviews saying that chloroquine should be continued to use, but somehow it is not able to get a very good foothold worldwide. Now I am summarizing the number one stage, that is membrane fusion of the virus with the host cells, where it's able to inhibit cell entry of the virus, and beginning with the serine protease inhibitors, which inhibits TMPRSS2, so that it should prevent binding of the virus spike protein to the ACE receptor, is camostat, nafamostat, which has already been approved for use in Japan but not in the US. And recombinant human ACE2, it competes the virus to bind. Of course, so far it is not being approved anywhere. Angio-17, it is hematopoietic stimulant. It is there for last several years, but nothing happened about this one not being approved. It is only remained only as a theoretical compound. It is, and ARBs is already approved antihypertensive agents, but you cannot take it as a drug. It is only people who are already been on therapy should continue, but other people should not be prescribed to other people, because normotensive people should not take it. So it is not meant for them. It is only people who are already been on this drug should continue to take it. Chloroquine emergency approval was there, but it was withdrawn in August, September. It was withdrawn only for 3-4 months. It enjoyed emergency use authorization. It is an anti-malarial, rheumatoid arthritis, this one. And Arbidol, it is influenza virus, respiratory virus, again approved in the Japan. Arbidol, it is a broad spectrum antiviral. It inhibits the membrane fusion. Now we'll move to the translation part. After attaching to the ACE2 receptor, it is endocytosed. Now it is going to replicate in the body by moving on to the ribosomes. There it is able to release polyproteins and as well as it is enzymes. So using host cell protein translation machinery, the viral translate into viral polyproteins, that is PP1A and PP1AB, and as well as the protease enzymes CL-Pro and PL-Pro. Plus it also forms RNA-dependent RNA polymerase, an important enzyme which is non-structural protein number 12. So protease process thus can be inhibited, so that if it inhibits this one, it cannot assemble. So sequence of steps should be inhibited by number of drugs which will be seeing this one. As we covered up to the hydroxychloroquine part here. Now this puromycin inhibits that at the stage of ribosome part, but not very effective. A lot of compounds, even by using CAD, people are coming up with thousands of compounds, including natural products from Indian laboratories. People have published a lot of papers on this one. Protease enzyme inhibitors as a polypeptides PP1A, PP1AB acting and should be breaking down by this proteolysis enzyme 3CL Pro MPro, and none of them may be able to withstand the virus ferocity. Lopinavir, ritonavir are the traditional ones used for HIV and Zika virus. They have been shown in laboratory everything, but clinically they are not that proved to be very effective ones. Remdesivir is able to block this application along with the favipiravir. They use a single as a combination, and they demonstrated that it is very effective to some extent. But of course, in India, we have seen that people are using remdesivir and out of stock. People are shown in black market. Other thing again coming up, and now generic ones are also coming up. Earlier it is a very costly one. Now it has price has been reduced drastically. Still, people are in favor of remdesivir. But only thing is a minor. It is not that very major effect. But from emergency authorization, it has been regularly approved. Now for the treatment of this, is the only one which has found approval, and then it is released to the exercise. This releasing part, we don't have anything which can prevent the release. Once it is being formed, it bursts out and gets released into the circulation. So remdesivir is the one which I little bit talk about it here, because currently everybody uses here, and it prevents the viral proteins. This somehow with this slide it doesn't come out the picture, which Time magazine very nicely published this one, how the proteins should be inhibited, that part. In the milestone, you see that in the 29th April of 2020, NIH trial suggests the benefit of this particular compound, remdesivir, and in 1st May, FDA gives emergency authorization, and then solidarity results comes out, and solidarity result from a WHO trial shows negative one, but Gilead trial shows positive one. There is a mixture of results. It is not all the trials results were not positive. Still they went ahead, and then finally it was approved in 22nd October 2020 approved, and beginning May was the emergency approval, and WHO solid result was negative actually. And only thing is it lessens the time spent on oxygen from 15 days to that of 11 days. 4 days it cuts down. That is not a great thing, people say. That means those who are in very much high titers and very much in pathetic situation, possibly it might work, because average 5 days sooner the patient will be discharged. For this way, people are spending thousands of rupees, may not be worth. People say that one. However, it is some people dispute the use of remdesivir for the 5 days, but the 4 days also very important. When a person is dying, definitely it comes as a very handy drug. So it is being much more favored. And ivermectin is also in picture, saying that in the later stage, it prevents the replication at this spot. Now, hydroxychloroquine acts here also in the packaging and wire assembly, which is more or less a theoretical one rather than practically. They are shown in the laboratory stages, but in the patient, in the clinical part, they are not able to demonstrate this aspect other than showing in the laboratory dishes. Now, summarizing this second part, inhibition of viral replication, transcription, translation, and release. We had HIV protease inhibitors, which are being used for HIV, that is ritonavir, leponavir, darunavir, and cobicistat. These are the drugs which are still trials are going on, but no emergency use authorization for any of these drugs so far. Remdesivir is the one which I put in the red one here, which has earlier been used for anti-Ebola, EUA. Now it has become a permanent one. Favipiravir for influenza. Then Tempol, which is a very important one. A lot of the people are using experimentally, which is a superoxide dismutase pimetic. It is a radical scavenger. Now, in the RNA polymerase 1, dependent RNA polymerase, they have shown that iron and sulfur clusters becomes a cofactor for this one, and Tempol has been shown to inhibit these clusters, and it has gained a lot of foothold in the recent trial, and also papers are published here show that it is much more beneficial when Tempol, and they should be explored further one. Still, it is an experimental investigative drug, and it has been being used in the laboratory for so many years. Now this has been extrapolated whether it could be used because of recently finding as an inhibition of the cofactor necessary for this RNA-dependent RNA polymerase. Other one is important one is inhaled nitric oxide, which is a very important one, which has also been approved. There are even generic ones are available, inhaled nitric oxide, for increasing the oxygenation in the respiratory during respiratory failure. You know that it is a gasotransmitter, it is a vasodilator, is S-nitrosylation of the viral proteins and thus inhibits the RNA synthesis, is an antiviral. And nitric oxide is earlier it was known as endothelium derived releasing factor. We know that 3 scientists from the US, all of 3 of them, Robert Furchgott, a famous pharmacologist from the State University of New York, then Professor Ignaro from San Diego area in the California, and third case is Fried Murad from Virginia. All 3 scientists are able to get Nobel Prize for the discovery of nitric oxide, showing that it is a vasodilator, very important because it is a cyclic GMP stimulant, and also it is beneficial in the heart failure disease. And one can generate nitric oxide in the body also. When you inhale air, when air able to stimulate the sinusoids present in the nostrils that interacts with the sinusoidal cells and generate nitric oxide, and when you take it inside inhalation, it gets into the lungs. So it's a very beneficial. But when you exhale, don't exhale through the nose, exhale through the mouth. Same thing we do in case of breathing exercise, pranayama, and also when you do with this yoga, when you say that Om or I, U, Om. So when you say that, you inhale the air through your nostrils, and when you utter those 3 words, then you use your mouth to exhale the air. So it's a very good one. This is what Ignaro, which is recently wrote in the British Journal of Pharmacology 2020 December issue. He writes, he doesn't mention anything about the yoga or anything else, but he says that it is always worthwhile to inhale and generate your own nitric oxide in the sinusoidal cells of the nose, but don't exhale through the nose, because whatever you generated, you are throwing it away, but exhale through your mouth. That is way you conserve your nitric oxide, that is able to kill the virus, and you store the nitric oxide. Of course, nitric oxide cannot be stored. You know very well that it is life cycle is only about, I think, 6 seconds to 8 seconds. That's all. It's very difficult to even do the experiments in the laboratory. It's very fast reaction. Nitric oxide generates and dies very fast. So that's why in the body it continuously generates. You keep on, because you also inhale air very quickly. You inhale, and when you keep on inhaling the air, you keep on generating more and more nitric oxide, and more keeps on entering your cells. So in case of mild COVID, people who are attacked in initial stages, tell them to do this breathing exercise, and so that they can be able to have more nitric oxide. This is what 2-3 papers have said, and especially it came back up by Professor Ignaro, who is a Nobel laureate, discovered this nitric oxide, was one of the 3 Nobel laureates in that nitric oxide cycle. Now, sirolimus is rapamycin, which already showed that it is immunosuppressant. It also inhibits replication, possibly at the transcription level. Other one is ivermectin, which a lot of people in India also people are using ivermectin. It is anti-parasitic, It is to treatment of diarrhea caused by the Giardia cryptosporidium, and suppresses the viral replication by inhibiting the maturation of the viral hemagglutinin, which is one of the structural proteins which I did not mention in the beginning because it's not that very important out of these 4 structural proteins. Now, with the intervention of the defective virus is produced, say a piece of chopped ones. So if it interfere with this one, RNA dependent RNA polymerase, then you will produce a chopped ones of the virus which you cannot assemble, cannot release it. Now, after the first one, we are moving to the last part, that is the host immune response. Now, the virus has entered the body, replicated, and it now released into the circulation, and it is inundating more and more cells. When it goes to the lung, macrophages, they interact and they triggers the immune system. When it goes berserk, it is able to create havoc in the body itself. Its own immune system turns against the body. That is the inflammatory pathways sets in, that is causing pro-inflammatory cytokines to be released in the body. Now I will give some introduction to this one, cytokine storm. A key feature of the SARS-CoV-2 mediated pathogenesis in the COVID is that it infects the macrophages and T cells, I showed in the beginning slides. When the virus attacks the lung cells, it infects macrophages and the T cells. It increases the neutrophils, reduces T lymphocytes. Of course, we know that in any infection, there is an increase in neutrophils, a part of the WBC defensive mechanism in the body. And because of this one, the IL-6, interleukin 6, which is a inflammatory cytokine, and this, any titer of this becomes a marker also, IL-6. They measure it. A higher IL-6 shows that you got set in the cytokine storm, and also C-reactive protein, that is the CRP level, which also shows that your heart system is becoming weakening. The inflammatory response to virus is excessive as a result of viral infection, strong or overactivation. This is the one which kills the people, overactivation immune response, which sets the cytokine storm, and this has to be antagonized by giving immunomodulators. So cytokines are regulators of host immune response to infection, immune responses, inflammation, and trauma. These cytokines are produced when innate immune system, that is our body, inherent one, natural immune system responses are activated. So that becomes to be in a mild form, but they become much more intense. That becomes the worst pro-inflammatory ones. And that's why we got pro-inflammatory cytokines produced, which prominently activating by the macrophages and helper cells are involved in setting up the motion of the inflammatory reactions. And this is a very beautiful diagram drawn by this one Indian article, Kumar. It came up in that year's open research, very beautiful diagram they drew, showing that which may be possibly difficult for you people to understand from the slides here because of small in nature possibly. So you see that the pathways are shown lymphopenia and T cells exhaustion, and sets in the increase in the neutrophils, as I said, and also granulocytic factors, which sets in the cytokine storms, which is IL, interleukin 1 beta, 2, 6, 7, 8, 10, and also colony stimulating factors, and TNF, tumor necrosis factor alpha, which are also triggered by angiotensin 2, and this should be combated by immunomodulators. See that this photograph came very nicely in the Science published by Weidman in the last year, showing that a ground glass phenomenon, they use a word, it looks like a ground glass, the whole lungs, you see the yellow spots and eaten away all the lungs. So you cannot expect a person to survive with having a ground glass phenomena. Oh, this man who died at George Washington University Hospital in the US, and they showed a 3 model picture of this lungs inundation. And see the clot. And the blood clot takes place in the blood vessels. This is because of endothelial dysfunction taking place. And earlier it was not shown in the heart, but later part of the 20th 20, they showed that heart and blood vessels are also being this one, and promotes clots and heart attacks and cardiac inflammation by this virus particle. Now, when this cytokines and chemokines sets in motion, and this large number of anti-inflammatory and immunomodulatory drugs are being prescribed. And the beginning, what they thought one of them was the user was corticosteroids. In last year, it came, and also the prednisolone, methylprednisolone was the one which various people thought best one to use this one, corticosteroids for combating this cytokine storm. Meanwhile, they also come up with the other immunomodulators, baricitinib, along with remdesivir should be used, not alone, otherwise it gives a lot of reactions. And sarilumab, tocilizumab, ivermectin are some of the drugs which physicians in each country they are prescribing to combat this cytokine storm. Now I'm summarizing this. What are the drugs which targets at that time when it sets in the hyperinflammation interfering with the cytokines? This anti-inflammatory compound, tocilizumab, sarilumab, and these are all MAB, ending with antibody. These are all being used in rheumatoid arthritis and also Castleman disease. These are being now repurposed to, but none of them has got any emergency use authorization. Still off-label they're using it, and also trials are also being going on. Again, anti-cancer substances like ruxolitinib, baricitinib, which inhibits JAK1 and 2 and antagonists interleukins and interferon N, and they're also being prescribed for rheumatoid arthritis. Other one which borrowed from arthritis, anti-arthritic drugs which can neutralize 1B was canakinumab, which is also being used by different countries. One, and thalidomide, you know, all this thalidomide tragedy took place in 1960s when pregnant ladies took this one, and thalidomide, which is teratogen substance. Now it is being approved for leprosy and multiple myeloma, which is impaired the synthesis of TNF alpha, and this way again trials are going on in thalidomide and should not be used during pregnancy. Corticosteroids and anti-inflammatory glucocorticoids, these are all corticosteroids. Methylprednisolone and dexamethasone is the one which is little bit scarcity was there. Dexamethasone being prescribed, and which inhibits TNF alpha and NF kappa B activation. Colchicine, which is an anti-gout, anti-cancer, being prescribed for the gout. Also this anakinra, which is IL-1 receptor antagonist, which targets the receptor prods, which also block the activity of interleukin-1, which is a rheumatoid arthritis drug. Remember, none of these drugs are being authorized by any of the regulatory agencies. They're all being, no emergency use authorization also, no EUA. Still they're being used as an off-label compounds for targeting this inflammation. Now, last part is neutralizing monoclonal antibodies. They are being approved. They are not part of repurposable drugs, but there are laboratory-made ones, and a number of companies are involved. What is this? Neutralizing antibodies have been detected in convalescent patients. These antibodies will attach, which I will be showing in the next slide. They attaches to the receptor binding domain of the virus. They indicate humoral responses are mounted rapid in the COVID patients. Antibody-dependent enhancement has been proposed as a mechanism to exacerbate SARS-CoV-2 infection. This is the same phenomenon used by the vaccines also. There are about 19 monoclonal antibodies targeting the virus spike proteins. Initial clinical trials, 27 completed the preclinical phase, and 49 are slowly discovery phase. Few neutralizing antibodies have been gained emergency use authorizations, which I will be showing in the other slide. Now, UAC maneuver, see spike protein, and this targeting this one, and this antibody goes and blocks the spike protein area. And if there's a mutation is there, we know that in India we are getting much more delta mutations. It is not able to bind this one, the antibodies. This is what they predict. So you have to say that those able to, in spite of able to bind, this would be nice for neutralizing antibodies. And what are these antibodies which have got emergency use authorization? These are the ones. RegenCov is the much more in the news when Trump was given RegenCov when he was admitted to hospital when he got COVID-19. Is made up of two the monoclonal antibodies which are administered together. So otherwise, we got 1, 2, 3, 4, 5 of them have been authorized for the treatment of the patients. And they remember, they are not repurposed one, they are laboratory made one. They have got a number of them are being prepared by a number of companies and released now and then. Yeah, I summarize this talk. I'm coming to the last part of my this one talk. Now I'm summarizing. Be ready with some of the questions if you have got anything. Now, several drugs are being developed and clinically tested for treating COVID-19. The clinical trials are based on the viral life cycle, which we discuss at various stages, 1 to 5. At different stages, we are able to step in and able to inhibit the next step from the host cell, at beginning with the host cell attachment, then entry, then to translation, replication, and release, and finally damaging our immune system. Thus, there are multiple targets one can be attacked for a potential therapeutic intervention. Now, repurposing of existing drugs can potentially be an efficient strategy for drug development. Drugs under consideration that could have activity for COVID-19, such as antivirals, protease inhibitors, antibiotics, statins, RAS inhibitors, corticosteroids, antirheumatics, antineoplastics, anti-inflammatory immune modulators, and anesthetics are also being used. But remember that all of them are only palliative, not curative. We know that the definition of drug itself is they are only for, even though they are meant for treating the disease, but they do not treat the disease completely. If you're taking antihypertensive or for heart failure or for the diabetes, you take for the lifetime. If you stop it, again it will come back. So that shows they are only palliative nature, no curative, only anti-inflammatory compounds, anti-inflammation which you take it, analgesic ones are antibiotics, they are only curative. You would get rid of the disease, the bacteria or virus, whatever is there, it will go away from the body when you take antibiotic. But not any of these antihypertensives, or when you take this for the diabetes one, they're only palliative. Don't be under the impression drugs are not miracles to have a curative effects. They're only palliative nature. They comfort you. Patients may be affected by SARS-CoV differently, and that many factors influence the outcome of the disease. The rapidly changing landscape of clinical trials may result in withdrawing from the clinical trials and may be successful, may not be successful, as we have seen in the worldwide. A lot of trials started, they dropped in the beginning itself, or because of some not able to show proper efficacy or safety, such patients, because they might have been withdrawn. So there are two mantras for any drug to be successful. One is the safety, another one is the efficacy. Safety and efficacy, they go together. That's why we've got phase 1, phase 2, and phase 3 trial, where it begins with the safety and ends with both safety and efficacy. So both these parts are very essential in approval of a new drug. 7 databases have provided dedicated information and drug repurposing database, and this British Journal of Pharmacology gives a full description of these targets. That is a database listing the drugs. The drug bank is a much more PubChem therapeutic target database on clinicaltrials.gov. Because if you do a search base, you will get the drugs which can be targeted for the disease. Now, the famous sentence, we know all this thing with Sir Isaac Newton started with standing on the shoulders of giants. Now, the future is not standing on the shoulders of giants, it is standing on the shoulders of crowds. It is a crowd hunting, a herd immunity against SARS-CoV infection is a community effort or collaboration. You have to reach certain target to get the herd immunity. Each one should reach one for the vaccination. So meanwhile, we have to stay healthy, follow COVID protocol all the time, so that we should be able to get vaccinated. Once we reach even if 40 to 45%, we should be able to decrease the incidences as well as the death also. Thanks for your time in attending this lecture. Thank you so much. Appreciated.
Thank you so much, sir. That was indeed very, very insightful, especially considering the fact that we are going through the pandemic and everyone wants to know more and more, wants to understand if there are no drugs, why are there no drugs, wants to understand if there are vaccines coming in at a faster pace, how are they coming at such faster pace? There's so many questions surrounding, so that was really insightful. We have a few questions, sir, if you don't mind. So the first question is, sir, for repurposed drugs, what are the additional and modified tests and trials that are required by FDA for regulatory approval?
First is the clinical trials that show the good result. For any agency, whether it is the Indian Regulatory Agency or the EMA, so what we need at present is a successful clinical trial demonstrating its efficacy along with the safety of this one. If the safety is not there, statistically significantly shown that it is effective, then only it gains approval. If there is no, that's why I given already instance about chloroquine, chloroquine trials. In some number of trials, they did not show. Even though practically people are treated and they showed benefit, but in large trial, when they compared with the placebo, it did not give that much benefit. In one trial it showed, other trial did not show, and also it's how much statistical importance the data is able to command, and that only leads. Again, second thing is it also should demonstrate a safety purpose. The compound should be more safe enough and be given to the patient. It should not untoward effects. So these two has to be balanced, safety and efficacy. Both are the two mantras to gain any approval of any drug by any agency in the world.
Yes, yes. Of course, I will come back to that point again. So the mantra that you've said about safety and efficacy and how it is actually effective, we can see. Before that, there's another question where the audience is asking, can we predict the PK/PD in silico while repurposing drugs?
Yeah, PK/PD, that also, when they are doing with that result is very much essential for any approval of any drug. PK/PD, they have to show a response. How the response in a curve manner, because they show it, that is, there is a linear response or anything else. Even for example, say for example, they're giving 500 milligram, the chloroquine is given. In 500 milligram, what is the titer value in the clinical pharmacology data, plus what is its effectiveness? So this PK/PD, even if you are doing a very brand new investigative drug, the phase 1 trial is a PK/PD in a healthy volunteers, they show it. In phase 2 trial, they show PK/PD response with using a single ascending dose, multiple ascending dose, they're able to demonstrate. So this PK/PD forms a part of the approval process.
Great, sir. So now slightly touching upon the mantra of safety and efficacy, that looks to be one of the biggest reason why the common public is abstaining from vaccine if they are, right? The question is like, generally it used to take 10 years, 12 years, and now within 10 months we have come up with vaccine. Is it safe? Is it efficacious? More than efficacy, I think safety has been a major concern for the general public. What is your view on it? What would you like to send as a message out there?
Regarding vaccination?
Yes, sir. Yes, sir.
Yeah, because a lot of concerns about the vaccination throughout the globe, globally. It is not only one. Even in the US, you have taken Pfizer and Moderna. Now they have put a warning, especially in they are given the college students now 12 to 16 years. They started this one. They could find some inflammation in the heart, and not that very high cases. About I think yesterday or I read something about 60 cases of inflammation. This was first discovered, noted in Israel, and now this causing mainly in the young adults rather than in the old people. that number is less. Now they come up with a labeling one. Yesterday or day before they announced it, and both Pfizer and Moderna vaccine saying that it may cause inflammation of the heart, and one should watch it. But of course, this is a reversible one. It goes away. Now with AstraZeneca, this one, and also with Johnson and Johnson, they showed some blood clots and happening with the clots. And again, yesterday one more I read it that after 3 weeks after vaccination in Australia, possibly a lady taking AstraZeneca vaccine, she died because of blood clot. So yeah, that might happens one and there, here and there. Yeah, if you make question, ask a question, what happens if the lady has not taken the vaccination? Don't you think she would have survived? Yes, she would have survived. And so that means, can we link it to vaccine? Again, people do not want to link it to vaccine. So in my opinion, had the person not taken the vaccine, they would have survived. Because of taking vaccination, they got the blood clot and they eventually died after 1 week or 2 weeks or 3 weeks. That causes hesitancy among people. But certainly, it should not be linked to the vaccine, saying that these vaccines causes any blood clots. And there could be some underlying disease, underlying comorbidity in these people, which might have hastened by taking vaccine possibly also. This is what some of the people have put forward, the hypothesis, why it takes place in few individuals, why not in all the individuals. That means the percentage is 0.01%. For their sake, you cannot shun away from the taking vaccination. Otherwise, the disaster sets in. For example, in India, we got in the recent 1.2 million deaths. Had all these people have taken vaccination, you would not have seen death of all 0.2 million. You would have seen something like 1,000 deaths, that's all, because of the virus. That shows that you would have saved more lives by taking vaccine rather than not taking the vaccines.
Correct. So, and it also has a lot to do with how the virus is, like you had also mentioned in your presentation, that SARS-CoV-2 is affecting different people differently, right? That's the reason the trials, that's the kind of challenge also the trials are facing, like you mentioned in your summary. Also, sir, so there's one more question where the audience is asking, is it possible to identify potential leads for SARS-CoV-2 through high throughput approaches?
Reword it again.
Is it possible to identify potential leads for SARS-CoV-2 through high throughput approaches?
Potential needs, what kind of needs you are talking about in this case?
Well, I like the audience who sent in the question to clarify. It's from the audience. Meanwhile, till we get a clarification, I'll quickly put in the second question, sir. Is there any web-based server for network-based repurposing of drugs?
Yeah, this is what I showed in the last slide.
Yes.
Web-based one that coming from the British Journal of Pharmacology. Last year, of course, you may search it, or if somebody has noted down that DOI number, they can search for that one. Drug Bank, and there is a BJP also publishes usually the drug database, which gives extensive list of the drugs which are repurposed ones, and even natural products. I think in India, somebody has generated that list of the things, what all the natural products which have been tried. Even the China, of course, China also is very large in plant chemistry, like that of India. We both are competing, actually, both China and India. In case of plant chemistry, a huge number of compounds they're shown in that drug design studies. Again, this all again is coming with either artificial intelligence or with a CAD. Of course, artificial intelligence is a part of CAD. They've shown that thousands of compounds are active on these sites. So you might get into the database. If you do some literature search, only last 2 years sufficient to see that one. You should be able to get both synthetic molecules as well as the plant molecules, which I don't know how much they have been purified, how much have been done. So you should be able to get hold of this one. Most of them are all acting on the as a protease inhibitors. Yes.
Okay. Yes, sir. So before we close for the day, we would really like to understand your take for the young audience who's listening to you and who look up to you. what is your message to them in terms of now coming back to the theme of the conference where we say 4.0, and we are trying to talk about how there has been a revolution in drug development field. So what is your message to them to stay ahead or to stay at the same pace with the kind of development or advancement we are going through?
Regarding the all drug discovery takes place at the university level. You know that most of the drugs come from the university, and once they demonstrate a good efficacy, need not be safety, because safety part lies with the drug company when they possess the drug from the university labs. So university labs should be devoted much more in the discovery aspect. So once you discover a drug, investigative chemical can be active on any one of this. For example, target, whatever you have chosen the target, we are screening for as a smooth muscle, intersmooth muscle, or a vascular smooth muscle, or on the heart, or for a diabetes 1 in the cells. So you should see that how effective the compound is now to combat, that is effectively produces a response. They should have a dose response. You should create, not with single dose, but in response, next dose it doesn't give any response. Not that way, as you already talked about PK/PD response. So same way you can demonstrate in the animals that you should be able to assay the concentration of the compound in the blood, and also when you give the drug, how far is able to, give orally. For example, if you give a compound and think you're given orally, 50% problem is already solved. The delivery problem also is very important. So if you're able to get a compound near to the delivery stage, and if a compound is able to produce a good response, then what will take up from the in vitro to in vivo study. And I've seen a lot of people do a short-term studies. Then if then that may not be good one. This cannot be done at a PG level, because in 1 year thesis work, people take a lot of trouble in designing a big study. They'll end up in problem, because procuring animals, giving drugs, and handling them, a lot of parameters are taken up. No, that is not possible. So you should not aspire to do a big thing at the postgraduate level, at M.Pharm level for 1 year. So take a small project, which your investigator, which your guide already knows, done with a previous student. Continue the same thing. A guide can give the same project for the particular drug to 2-3 students in consecutive years, so that it can develop into one stage to second stage to third stage, fourth stage. By the time 3-4 years, a lot of these have been produced, you would have got a very good molecule, and keep publishing that one. The result, we are doing a very standard procedure using in vitro cell techniques followed by the in vivo system. You got a very good results, which is worthwhile. Then you can investigate the toxicity of this one as compound, both acute and a short-term, long-term toxicity studies, 1 month to 3 months. You can do it if time permits. One student can do only toxicity, not the efficacy part. So other student can do only the efficacy part. So in this way, can divide. A PhD student can take the elaborate one, because I have valued some thesis for a ginger. The student had done a lot of work isolating a lot of compounds from the ginger and doing the chemistry also, then taking it to a pharmacological screening part, and both in vitro as well as in vivo demonstrated. But again, they did not do the PKPD part to demonstrate how effective it is in terms of concentration. That part is not possible to do it possible, because ran out of time. So one has to finish in 3 years. So I would specify that a postgraduate student should stick to a small project in deciphering the efficacy of a compound as much as possible, using a simple system, not going for a complicated system.
Thank you so much, sir. And I believe it does address a very important challenge that we do face. So if I'm to summarize on what you've just said, is to be devoted to discovery when you are being part of research, and to if possible, divide it into different aspects such that it can be continued even if the student is out of the college and it can proceed further. We look at a larger picture, we focus on discovery rather than just having papers or rather than just having research for the sake of it. That's amazing. So because yeah, that's one place where of course students also face, like you correctly said, they have a year or year and a half and they have a lot to do within that time, and of course everything does not become possible, and then we end up with having with average level of research as well. So a very
the project what you take at M.Pharm level, you can continue for PhD also.
Yes.
You need not necessarily under the same guide. You can go to another institute. You can say that, okay, I worked on this material, this ginger, for example, or curcumin. It is showing very good effect. All these things. I got this much plan. I will isolate the chemicals in this one, which has not been shown by others. I'll continue for my PhD work, and I'm sure that I should be able to back my PhD. This one. So, if the student, then the student can develop that into its own area, away from his M.Pharm and away from his M.Pharm guide, and develop his own lab. He or she can do it. So that shows that one can develop into a different arena itself. Though that means it becomes a M.Pharm is only a stepping stone, unless somebody doesn't want to continue research only 1 year and then go for another field. That is a different aspect. But if one stick to one field and taking this aspect, one can continue for the same project for PhD also, and then again apply for grant based upon this project, because already got a lot of data being generated. That becomes a background material which a granting agency always asks for it. What work you have done previously? A novice doesn't get it. Just like which comes first, seed or a fruit, a tree. So in the same way, even unless you publish something, nobody gives a grant. But without grant, you cannot publish anything because you don't have money. So you had to publish something at M.Pharm level, carry that work to PhD, publish more work. That becomes a basis to attract the grant as well as to get a faculty position or a research position in any of the any of labs or the pharma company.
Yes, completely in sync with our previous speaker who spoke about a gradual development of interest, rather than through influence. Like you said, what have you done in the past in that area that you've developed interest into and you're working further in? So thank you so much, sir. It does give a lot of insight, not just to the students, but also to us as academic institutions on how maybe we could guide our students further and design on research that are more discovery focused and more long-term focused. Thank you so much, sir, for being with us. It was wonderful to have you. And Professor Malia conveys his regards for this wonderful session, for your time. And of course, you'll be with us in panel discussion tomorrow. We'll meet you again tomorrow, sir. Thank you so much for being here. And any questions that are pending from the audience, we will also share with you through email, sir.
Yeah, tomorrow also, if you want, I will be there at your time, my time, 7 o'clock. Means it will be, I think I was asked to join around 5 o'clock, your time.
Our time, yes, sir. Yes, sir.
The panel discussion starts.
Yes, sir. Panel discussion starts at 5:10, sir. Yes.
5 o'clock. So I'll be logging at 5 o'clock.
Sure.
Okay.
Sir.
I'll
yes,
see you at 5 o'clock. Okay. Thank you so much.
Sir.
Appreciate it.
Thank you.
Bye bye.
Thanks.
Bye
bye.
Bye bye.
And now, before we call it a day, we cannot stop ourselves from expressing our gratitude towards the man who has been a pillar of support throughout. Our chairman, sir, Professor Dr. Suresh Nagpal. Krupanidhi Group of Institutions was started more than 3 decades ago. It was and it still is the product of the vision held by him, who dreamt of making top-in-class education possible as well as accessible to every desirous student. He is an educationist and a visionary who, with over 3 decades of efforts, has brought about a change of a lifetime in the education system. Sir has revolutionized the way education is imparted through the many colleges under the Krupanidhi Group of Institutions wings. His efforts for changing the way education has been looked at has been recognized with numerous awards time and often. One of it is the award for Academic Visionary Award for Excellence in Education, Leadership and Innovation in the Academicians category of the WCRC Leaders Asian Education Excellence Summit and Awards. Till today, Sir has left no stone unturned in developing world-class education system at Krupanidhi that houses students from over 35 countries now. Once again, sir, on behalf of the entire organizing committee, I would like to extend a heartfelt gratitude. Thank you so much for the support that you provide. Thank you for being our guiding light throughout, and thank you for motivating us always, sir. Thank you so much. And now, dear attendees, before we depart for the day, I hope you had amazing day today. The speakers, I was there with you throughout. We had amazing knowledge exchange. There were a lot of informations, and that too at such deeper level shared with us today. It is our honor and it is our luck that we were able to be with the kind of speakers that we had. Of course, we cannot thank enough our chief guest, Professor Dr. B. Suresh, and our chief patrons, our chairman, sir, Professor Dr. Suresh Nagpal, as well as Mrs. Geeta Nagpal, our vice chairperson. Before I close for the day, let me quickly give you an overview of what is lined up for tomorrow. So tomorrow we have 5 speakers lined up. We start again 1:30 sharp, and then we proceed with all our scientific sessions in this hall, while we will have our poster presentations running in the subsequent hall like today. Of course, that too was a great success today. We had lots of poster presentations. There was very good amount of question and answers that happened between the evaluators and the presenters. It was a very good energy over there as well. So tomorrow we have Dr. Mahadev Rao, who is our first speaker, followed by Dr. Sumit Maiti. Then a hands-on training by Dr. Murlidhara, for which we have also sent a notification. I'd like to emphasize again, please download the JMP software. The link for that has already been sent in your email IDs. That is essential, especially if you are really interested to be part of this hands-on training. Post the training, we have Dr. Venkatesh M. Joshi, and our last speaker for tomorrow is Dr. Tippaswamy, who again comes in all the way from Saudi Arabia. Post the speaker sessions, we start with our panel discussion at 5:10. The topic for panel discussion is Emerging Technologies in Drug Development, a Changing Landscape. The session will be moderated by our very own Professor Prakash V. Malia, and our speakers of day 2, along with Dr. G. Jagdish from today, will be part of the panel discussion. I hope to see you all tomorrow. Have a great evening ahead. Stay safe, and I hope you had a lovely day today. Have a lovely evening ahead, everyone. Thank you so much. See you tomorrow.